Vamos a dar inicio a la sesión del día de hoy y pues tenemos el gran honor de contar con la presencia del doctor Baltasar Becerril, quien, voy a dar una breve introducción, realizó sus estudios de doctorado en Ciencias Químicas en la Facultad de Química, entonces aquí es su casa, doctor, en donde recibió la medalla Gabino Barrera en 1988 por su destacada tesis doctoral. Actualmente es investigador titular PRI-DC y miembro del Sistema Nacional de Investigadores en el Instituto de Biotecnología de la UNAM en el Departamento de Medicina Molecular y Bioprocesos. Tiene 50 años de servicio académico en la UNAM y ha impartido clases en los distintos niveles de licenciatura y posgrado y ha formado un papel central en la formación de recursos humanos. Fungió como coordinador del programa de maestría y doctorado en Ciencias Bioquímicas entre el 2000 y 2004 y fue miembro del subcomité del 2021-22. Ha dirigido y cosupervisado numerosas tesis de licenciatura, maestría y doctorado. Tiene más de 100 publicaciones, muchos capítulos de libro, 14 patentes y pues su obra tiene numerosas citas y también, algo muy importante, participó en una transferencia tecnológica a los laboratorios Silanes. Entonces, welcome, Baltasar. Thank you very much for being here.
Thank you.
The floor is yours.
Good morning, everyone. Thank you for being here this so early. I would like to thank the organizers for giving me the opportunity to share with you this talk. Today, I'm going to show you how two biotechnological platforms, phase display and directed evolution, are expected to transform antivenom production, promising safer treatments and a future, hopefully, in which no life is lost due to scorpion sting. Let's start by introducing the problem and why innovation in antivenom production is so important. Scorpionism is an important public health problem in Mexico. In the last decade, an average of 300,000 accidental stings per year has been documented, caused by at least 24 species of the genus Centroroides, with 30 to 50 deaths. These statistics highlight the urgent need for the generation of an effective antivenom with present-day technologies. The symptoms of envenoming are caused by toxins that affect voltage-dependent sodium channels present in excitable cells. The binding of the toxins to the channels induces a functional impairment of nerve cells, which results in a blockage of nerve impulses and eventually death. How is scorpion sting currently treated? The treatment relies on an equine-derived antivenom in the FAB'2 format named FAB therapy, which consists in a use of a mixture of the venom from 4 of the most toxic scorpion species of the genus Centroroides, which contain most of the different types of medically important toxins. This mixture is used to hyperimmunize horses, and the plasma of the best responder is treated with pepsin to eliminate the FC fragment of the immunoglobulins, leaving the FAB'2 part of the immunoglobulin. The FAB'2 fragments are purified, assessed for their neutralizing potency, lyophilized, and packaged. Although this antivenom is very effective, in some areas of the country, up to 5 vials need to be administered when the standard treatment establishes 2. This observation could be an indication that the quality of the antivenom should be improved by including another venom in the immunization mixture. How is this problem approached in a modern way? By means of this disruptive innovation technology like phage display, which consists in the following. A blood sample from a healthy donor was used to separate B lymphocytes to prepare total RNA. This RNA was used for the amplification by RT-PCR of the DNA segments that encode the variable domains of the heavy and the light chains joined by a peptide linker constituting a phage antibody library, as I will indicate in a moment. The important is that we have a collection, a repertoire of recombinant antibodies in the single chain format that consists in the variable domain of the heavy chain to the light chain joined by a peptide linker, as already mentioned. This repertoire is cloned into a phage meat. A phage meat is a cloning vehicle with two origins of replication, one that works in E. coli and the other one is an origin of replication of a filamentous phage. The transformation of a culture of E. coli with this library and co-infected with a helper phage that is defective in replication allows to display the single chains fused to a protein at the surface of an infective particle named phage antibody. This collection is used to put in contact with immobilized toxins and for the identification of specific single chains that bind to lethal toxin from scorpion venom. The advantages of the single chain antibody fragment, among others, would be its rapid distribution and elimination from the body, specifically targeted against lethal toxins, improving potency, consistency, and reducing, more important, the amount of protein injected, and its easy handling at the recombinant DNA level. And here I show you the conjunction of both biotechnological platforms where if a selection process has been started, the specific binders are recovered and expanding in a fresh culture of Escherichia coli. This consists of 3 to 4 iterative rounds of incubation, washing of the non-binders, and the recovering to start a new cycle. Usually, it's necessary to implement 3 cycles of directed evolution, which means that the best binder is used to construct a mutagenic library, a mutant library to include diversity and subjected to new cycles of selection. The best binder, the final best binder, is expressed as free protein in E. coli for the determination of the affinity against toxins in a real-time molecular interaction sensor. And the most important is the evaluation of the best binders in vivo neutralization experiments that are of 2 types, mixing type or rescue type. In mixing type, the toxin or the venom is incubated with the single chains, incubated at room temperature around 30 minutes, and then injected into the mice. But the rescue type, which would resemble more a real sting, consists in the injection of the mice with the toxin or a venom, and let the real envenoming symptoms appear, and then inject the mice. Here I'm show you the story of all this. And at that time, when this started, we knew that there were only 7 deadly scorpion species. And this story started with the construction of a phage antibody library of around 100 million, and this library was selected against the most deadly toxin of the venoms, named CN2, from the Centauroides noxious species. After 4 rounds of selection, 2 main single chains were obtained, named 3F and C1. They were strong binding and specific against toxin CN2. With this picture, phase display and panning cycles with their iterative rounds yielded highly specific anti-CN2 single chains, providing lead molecules for further cycles of mutagenesis and selection. The lead single chains were strong and specific in binding, but were not neutralizing. So that led us to implement different routes of directed evolution against different toxins with resulting single chains indicated in bold: LR, RIS27, HB, NFG2, C1. Here, toxin CL13 was not recognized by the already developed single chains. This became a challenge for 2 reasons. One, this toxin is the second most deadly toxin. And second, Centauroides limpidus is the species of scorpion that is responsible for 10% of the total accidents. So we asked for a collaboration to implement hybridoma technology. We obtained a monoclonal. Its single chain version was obtained, and after 3 cycles of directed evolution, single chain 11F was obtained. Here I show you a cartoon of a ternary complex of this guy, RU1 from C1 root and LR from 3F root, complex with toxin CN2 indicated in red. And here the important is that neutralization experiments indicated that related variants in the C1 root requires nanomolar affinity to be neutralized, but in the other route it was worse. It needs subnanomolar affinities to be neutralized. With this picture, high affinity, epitope diverse single chain combinations are required for effective venom or toxin neutralization. Here I show you pictures of females and males of 13 of the 24 species, and due to the fact that there are at least 24 species of venomous scorpions, effective venom neutralization requires knowledge of the relative abundance and lethality of medically important toxins. Here and in the next slide, I show you a kind of catalog of the 24 species in terms of their location. In 12 of those 24 species, the characterization of their venoms in terms of the determination of the molecular weight of the medically important toxins and the relative abundance. And in 10 out of the 24 characterized at the level of which sodium channels are affected, and the lethality, indicated as lethal doses, 50 of 100%, indicating the amount of toxin or venom capable of killing 50%, 100%. Here, there are venoms with only one medically important toxin, but there are others in which up to 5 toxins are medically important. Here I show you the minimum and maximum lethality in LD, in this case LD50, corresponding to toxin CN2 with an LD50 of 250 nanograms. And the LD100 with 2.5 micrograms. Where I am here. I don't know what happened. Okay. Here, from the 12 characterized venoms, 30 lethal toxins were identified. Here, I show you the alignment of those 30 toxins where this is the levels of interaction of the optimized single chains, where the affinities are as low as 50 picomolar for the single chain LR for toxin CN2 to 6.9 for the single chain HV for this toxin. And for 10FG2, this is the range of affinities from 230 picomolar to 11.8 nanomolar. It is remarkable here the cross reactivity of this single chain. If we take into account that these symbols in triangles mean that the estimation of the affinity has been done, and we think that it's enough to be neutralizing. However, the neutralization has to be confirmed. But it's remarkable, the cross-reactivity of this guy. And the summary of this is that the optimized single chains interact with 16 toxins with affinities between 50 picomolar and 6.9 nanomolar. For 10G2, its range of affinities is between the mentioned values. Here is a summary of the neutralization of 16 venoms, and indicated in bold are 9 species that has been neutralized by a single chain or a combination of them. And here a concept important is, for instance, for LR or 10FG2, they independently are capable of neutralizing the venoms, but the optimal neutralization is reached with a combination of them, and it's the same for all these combinations. This is the minimum LD50 of the venom corresponding to the most toxic species, Centroides noxious, with 2.5 micrograms per 20 grams of mouse, and Scotoratus 22.7. These data and the ones from previous slides allow to conclude that single chain 10FG2 has the ability to neutralize 6 venoms, an estimated total of 20 toxins, being the single chain with the widest cross neutralization capacity reported so far. Here I show you the pictures of the 9 species neutralized, and this is the 10th one, whose name is Centroides mascota. The characterization of mascota venom has already been published. The manuscript on venom neutralization has been submitted for publication. The venom of Centroides mascota has 3 main toxins. Two of them has been fully sequenced, and the third one still needs to determine the last 4 residues at the carboxy terminus. Oops. Here, in the route of the directed evolution from C1, as you remember, up to 1052, 2 additional cycles of direct evolution were implemented to obtain this guy, 2F. Here is a neutralization test with 3 LD50s of the venom of Centrorides mascot, both mixed type and rescue type. In the mixed one, 1082 was capable of protecting the whole, all the mice, but in the rescue assay, only 33%. What does it mean if we see that a combination of 10FG2 plus 2F recovered the protection and compared with the control where all the mice died? It means that these results would indicate that a solid neutralization criterion should include 3 LD50s of fresh toxin or venom under rescue conditions. That would be here. I show you a cartoon of the structure of 3 toxins, just to show that these 2 toxins are very similar in the regions indicated by the black bars that are the points where the single chains contact the toxin. These are very similar. They are neutralized by the obtained single chains, but C13 is divergent. That's why the single chain didn't recognize it, and that is our commitment to get variants capable of neutralizing this toxin, 3LD50, in rescue condition. Here is an alignment of toxins, in this case, really, in relation to CSS2, where we can see that all this group is very similar. Here turns to the divergence is, is clear, but here with this group where it's CL13 and other toxins, we name this group of toxins similar to toxin CL13, and it's the commitment to obtain variants, whatever. For this, and knowing that there are 24 species of venomous scorpions, if the average number of lethal toxins per venom were 3, we would be facing with an enormous challenge of neutralizing 72 toxins. In the directed evolution from the monoclonal to this guy, here we knew that combining 11F with 10FG2 in the best of the scenarios is possible to neutralize 2 LD50s, but not 3 LD50s of Centroroides and then. After several rounds of directed evolution, variants 1B, 1BG were obtained, and these are the results that I would like to first say here. For the characterized venoms, the amount of toxins present is known, which allows to calculate the amount of single chains required for neutralization, so that for each molecule of toxin, there should be 10 molecules of single chain, and this is the control. This is a mix with 10FG2 and 1B, protection 66%, but the mix with 1BG protected 80%. With this picture, I can tell you that variants from 1B are showing reactivity with good levels of affinity, hoping to have variants that could neutralize elusive toxin. The result of an experiment done here after I have sent my presentation is that raising the proportion of single chain 10Ab2 and 1Vg to 20 was capable of neutralizing 3LD50s or Centroides limpidus in rescue type of experiments. Finally, looking for alternative sources, we decided to look for chicken bodies to have variants to have the chance to neutralize the elusive toxin. This is an overview of a 6-week chicken immunization schedule using CL13, CV2, and CHO5 toxins with increasing doses of adjuvant, including periodic bleeding and final sacrifice. ELISA results show that strong antibody responses with CL13 and CV2 induced higher titers than CHO5. After 5 panning cycles of the chicken library, around 20 million variants, the best binder was this one. This one was mature with 1 cycle of directed evolution. We obtained 3 variants, and for 6D, showed the strongest improvement. Kinetic analysis revealed a marked affinity enhancement for CV2, where the KD was reduced from 53 to 5.6 and improved the binding to CL13 toxin, hoping once again to have variants that could allow to neutralize elusive toxins. Finally, human single-chain LR and 10Ab2 were expressed in Pichia pastoris, achieving up to 12-fold higher yields, around 50 milligrams per liter, compared to periplasmic expression in E. coli, around 4 milligrams, while maintaining identical binding and neutralization properties. And when this, the conclusion would be that phase display and directed evolution enabled the development of high affinity single chains capable of neutralizing 10 Mexican scorpion venoms and other toxins, paving the way for the scalable production of a recombinant antivenom using bacteria or yeast grown in electronically controlled bioreactors. This strategy would eliminate reliance on scorpions and horses, reduce animal testing, and ensure consistent, reproducible antivenom quality. Finally, I'm grateful to Dr. Lidia Viaño, who has been collaborating in the entire development of this research line, to Master Sciences Timoteo Lamendi, Leopoldo Boreca, Carmen Flores, Alejandro Olvera, Luis Fernando Lozoya, Doctors Ernesto Ortiz, Ilse Gómez, Guillermo Fernández, Alberto Moreno, Hugo Valencia, Bachelor of Sciences, Natalia Baena, Emilio Jiménez, and many other students and colleagues who have contributed to the results. So Dr. Posani and his group, to Public Accountant Maria del Carmen Martinez, to CONACYT now City, for the grant to RAI 3045, whose technical responsible was Dr. Alejandro Laguna. Thank you very much for your attention, and questions, please.
We have time for some questions.
Yeah.
Okay, so thanks for your really interesting presentation. I really enjoyed it. My question would be regarding the directed evolution approach. So you make a round of directed evolution, have a lot of different mutants. Are you only taking the best mutant and then going to the next cycle? Or are you, for example, taking like the 3 best? And when do you decide to stop the directed evolution cycles?
Yeah. Determining the affinity of the single chains against the toxin is a criterion. When we reach certain level of association and dissociation, the relation of dissociation against association is the KD. When we reach some values, we suppose that they should be neutralizing. But sometimes, after neutralization tests are done, it is not, it's not what we expected. So that variant is subjected to another cycle of directed evolution.
Okay, so you're testing after the cycle, more or less, it's already where you
At the beginning, it was relatively frequent to do that. But now, with this criteria, most of the times is successful in neutralization.
Super, thank you.
No more questions? No?
Yeah, thank you very much for the talk, very interesting. My question was about kind of the more practical aspect. So if someone is stung then by one of these deadly scorpions, how soon after being stung do they need to be administered with this antivenom in order to recover? Because in the mice studies, you waited for the mouse to show signs of suffering from the venom before then administering the antivenom. But for humans, like how soon after them being stung do they have to be administered, or else too late?
Okay. Of course, there are criteria among medical personnel specialized in attending stung people. But let me tell you that we not have only used mice as a model animal, but also lambs, but of about 15 kilograms, which resemble more, and the symptoms are reproducible between mice and sheep. Okay. And for children, it's recommended not to last more, I would either say that 45 minutes to take the children to the hospital. For an adult, it could be a little longer, but no more than 2 hours.
But because the places where people are actually being stung by the scorpions, is it in the city, close to hospitals, or is it far away where you have to travel really far to get somewhere?
It depends. There are people that believe in home remedies. The indication, especially for a child, is go to the hospital. And an adult could resist, but the recommendation is for both, especially for children. Okay, thank you for your attention.
One quick question. How big is the market of these antivenoms? And also, I believe that the US scorpions are the same as the ones in Durango, right?
Yeah.
So the antivenoms are made in Mexico and sent to the US, or they have their own companies?
The antivenom used in the United States was developed by a pharmaceutical company from Mexico, Laboratorios Silanes. By the way, Alejandro Alagón helped to develop this orphan drug that was accepted for DA in the United States. Actually, we have a mix that neutralizes the species in the south of the United States, and the market in Mexico is 600,000 vials per year.
Wow. Okay. Thank you very much. Cuéntanos qué. ¿Que dónde están los alacranes vuelos?
The color is relative. Usually people say that if the scorpion is güerito, it could be deathful. Not really. There are very black scorpions that are also deathful. They are distributed all around the side part of the Pacific, even in the center of the country. For instance, where we live in Cuernavaca, in the surroundings of the Institute of Biotechnology, there are deathful, I mean, the species that causes the 10% of the total stings.
One last quick question.
It's a really quick question about the
I don't hear you.
Hi, yeah. It's a question more regarding the bioprocess part, and it's like, you know exactly how much of these antivenoms you can produce now that you're like scaling up into the bioreactors, or is just like in a raw phase right now,
Yeah.
or
Actually, I don't have the answer, but I can tell you that if you have a fermenter of 500 liters, that would be enough 2 processes per year, hoping to have a yield of at least 20 milligrams per liter. But if you use yeast, it's easier. It's maybe a single preparation in a 500 liters fermenter would be enough to cover the needs.
Okay, well, let's thank again Dr. Becerril for this wonderful talk. Thank you very much.
You're welcome.
Sí, ahorita sí.
My computer's okay, Mario?
Yeah, sure.
Will jump drive too, man.
Do you have it has a HMD or the living one? Okay, that's good. The old one.
Yeah, and just basically.
It's working. Let's see.
I can just use mine. You can just use the thing. Use mine. I have a thing.
Oh, you have your thing? Okay, okay. Let's see, just to get this.
In the presentation mode.
Yeah, it's. There you go.
Thank you, man.
Oh, it's out, or it's a little bit out.
Oh, is it bigger? That's why, maybe?
Yeah,
Oh,
it's fine. I think it's fine.
Yeah, it's okay. Yeah.
Bueno, nuestro siguiente invitado es el doctor Josefa Raya, quien es investigador asociado de la Universidad de Carolina del Norte en Greensboro, en el Departamento de Química y Bioquímica. Él hizo su doctorado en biología con especialidad en micología en la Universidad de Illinois en Urbana-Champaign en 2007, y luego de una estancia postdoctoral en la misma universidad se incorpora a la Universidad de Carolina del Norte, en donde de hecho estuvimos juntos muchos años, en donde investiga la diversidad y biología de los hongos. Uno de sus proyectos principales se centra en los metabolitos secundarios bioactivos de hongos, saprobios, incluidos los hongos acuáticos, y quien es reconocido mundialmente por el estudio de estos organismos. También realiza colaboraciones interdisciplinarias con numerosos países sobre biología fúngica, taxonomía y sistemática molecular. Entonces, welcome Josefa, this is your home and the floor is yours.
Thank you, Mario. Thank you very much. Thank you, everybody, for being here today. Thank you so much for the invite. I greatly appreciate it. I just realized that I have a white screen on my PowerPoint, so a few slides might be out of the image, but I think we should be okay. So I'm going to talk about freshwater fungi and what we are doing with these fungi and their chemistry. So the outline of my talk today is why we study fungi. Why are we studying ascomycetes? So I'm going to talk about what ascomycete fungi are, who they are, how to find them, what do they do, why we study them, and what have we discovered from it. That's basically the premise of my talk today. Just a timer for myself. So So I'd like to show this slide. I really like this slide because it sort of gives an idea of the modus operandi of our lab. The first part of our work starts with me in the laboratory and in the field, where I go collect samples, bring them back to the laboratory, isolate them, purify them, and identify them. I'm a fungal taxonomist by training. I really like to see a genus and species name on things. So that's one of my expertise. And I'm really interested in finding novel diversity, because in my mind, novel diversity is what leads to novel chemistry. And so then I work with Nick Oberlies at UNCG and his lab, where they are studying the chemistry, right? So we get the fungus, we get it to grow, we isolate it, purification, all the chemical methods, spectroscopic methods, NMR methods to identify it, and then we do bioassays. We also do bioactive guided fractionation, so in order to sort of get to a molecule that has some treatment modality. So why are we studying fungi? In the fungal kingdom, as you see, is extremely diverse. It is phylogenetically, ecologically, morphologically, and chemically very diverse. This kingdom is by far the second largest kingdom of life on Earth after the animalia and the insects, right? So its estimated number of species is supposed to be 1.5 to 5 million species. So there's a lot of diversity, and a lot of it is unstudied. So the 90% of the fungal kingdom is unknown, right? And fungi are seen in all kinds of different habitats, right? They are inside the rooms, outside. They are in plants, outside plants. They're decomposers, they're pathogens, they are human pathogens, they are plant pathogens, they are lichen associates, they are symbiotic associations. You can find them in rocket fuel, you can find them in Chernobyl nuclear reactors, you can even find them in the space station, right? So they are pretty much everywhere. And so this diversity is what we are interested in exploring. And our focus is the Ascomycota, and I'll try to get into that in a little bit. And so because of this unique morphological diversity, from microscopic yeasts to macroscopic mushrooms, we are interested in studying the fungal chemical diversity. And they make all kinds of molecules because they have the biosynthetic gene clusters capable of making this diversity. I want to show this slide here, mainly because this has the phylogeny of the kingdom. Sorry about the part that's got cut, but what I want to show you is that this is the kingdom fungus, and all of these are different phyla. And the ones that we are interested in are the Ascomycota, right? And supposedly the Ascomycota are the largest group of fungi and has the highest diversity as well. So, right? So these fungi are extremely diverse, and they make molecules of all kinds, apparently in the fungal kingdom, they are the most chemically diverse, and that has been proven from genomic studies. They have the most biosynthetic gene clusters within them to make novel chemistry. And this part here is very interesting because this group here, Pizizomycotina, again has the highest BGC diversity in the Ascomycota. The yeasts are also part of the Ascomycota, but the yeasts don't make a lot of chemistry. The numbers actually are showing the gene clusters and the capacity of these fungal groups to make enzymes and catabolic proteins. And the bracket numbers are essentially genome sequenced. Nick mentioned yesterday that 1 million bacterial genomes have been sequenced, but we have only access to close to 25,000 or so fungal genomes in GenBank. So we are still behind the other kingdoms of life. Right, this is just a diagram to show you that the ascomycetes have two cycles. One's the sexual cycle and one's the asexual cycle. And the sexual and the asexual cycle are both morphologically different. And that's a problem for chemists because sometimes you get the sexual cycle, sometimes you get the asexual cycle in culture, right? And naming is different for these cycles. So that's always been a problem. But now the mycologists are starting to put the sexual and the asexual names as one name so that the rest of the folks can also study them, right? So just want to show you that that always happens. And in the sexual cycle, the main thing is that it makes an ascus and you have ascospores. And then here you simply have conidia that are just shot out. So Aspergillus and penicillium is a good example of the asexual cycle. A lot of fungal drugs that have been in the market have come from ascomycetes, and a lot of them are from the asexual cycle. So it's interesting to see that, because you would think the hypothesis there, as far as I'm concerned, is because asexual cycle, the fungus is simply making spores. The rest of the energy goes into secondary metabolites. But in the sexual cycle, sexual reproduction takes energy, and sometimes maybe some of the sexual cultures of the fungus are not making as much chemistry, but that still has to be determined. So what are the freshwater ascomycetes? So these fungi occur on submerged wood and herbaceous leaf litter, deciduous leaf litter, right? So on the wood, you can find these fruiting bodies when you collect the wood and bring them back to the laboratory. They're microscopic fruiting bodies. You need a microscope to see them. There are all kinds of shapes and sizes. And then on leaf litter also, you find different types of asexual spores. And you can see the different shapes here. Essentially, those shapes are evolved to survive in freshwater habitats, right? And then you have this final group here, which are kind of terrestrial fungi that kind of get washed into freshwater. But there are some groups that are native to freshwater. This fungus here is an aeroaquatic fungus, and it happens to grow mostly in bodies of water that are standing bodies of water, like ponds and lakes. And what happens is, when the fungus on which it's growing, the substrate it's growing, comes to the air-water interface, these globular conidia detach, and then they go on and move and get new substrates. Right? So all of these are adapted to aquatic lifestyle. This is like a multipod. Imagine being a multipod and a tripod. You have a better hold on the leaf and it's in water, as opposed to a unipod. You would go straight to the bottom of the river or lake. Right? So these are different kinds of fungi that you see, and they are adapted for life in freshwater habitats. Right? These are all pictures. You can see they have gelatinous materials. These are spore modifications, and you can see these are hydrophobic large conidia that are modified for aquatic life. This is a very cool fungus. It is basically like gelatin on the wood. And this was called bullymyces because it's just a bunch of globular cells that help and float in the water. And here, I don't know if you can see, this fungus there's a balloon here, ascus, and that within the ascus there are all these ascospores. And then this is basically hyphae that are really thick and rope-like, which attach to wood, which is soft, sort of anchors themselves in the water. So they're all different kinds of modifications for an aquatic existence. Here are more pictures. I really love the morphology in these fungi. That's why I am in mycology, because of the diversity. I like to see them under the microscope, take pictures, and you can see all kinds of beautiful fungi, both from the tropics and temperate habitats that I've come across over the years. So this is an ascus with 8 ascospores. You can see different kinds of asci, and then you can see the ascospores have modifications of the sheath. This one was one of my favorite fungi from Ecuador. And then this one is one of my other favorites from Peru. So the tropics have really amazing diversity. So Mexico has probably the best diversity of freshwater ascomycetes. And Maria Gonzalez in the audience has been studying these fungi in Mexico. And I hope to collaborate with you in the future, Maria. Look at the diversity of these beautiful fungi. So how do we collect them? Basically go out in the field and get samples back to the laboratory and collect them. Sometimes you find gators near the water, so you want to be careful. This was in Florida, where I was doing field work when I was a doctoral student there in Illinois. So you collect the samples. The wood needs to be soft, so that soft wood indicates that it has got soft rot. And then you bring them in the bags like this with a paper towel to keep them moist. Bring them back to the laboratory and incubate them. And then you look at these fruiting bodies that pop over a period of 6 to 8 months. Once you see the fruiting bodies, you can singly isolate those fruiting bodies, get the spores in culture. I use antibiotics, so I kill all the bacteria. Sorry for the folks that study bacteria. I mean no harm. I am interested in the fungi, and if the bacteria are present in the media, the fungi do not grow because obviously they're slow growing, and so we need the antibiotics. Mostly we use penicillin G, streptomycin sulfate, to get rid of the bacteria, and then you get pure cultures of fungi with which you can identify them, with which you can do chemistry and all kinds of physiological work. So what are they doing there in the water? Well, they're important decomposers like fungi are. They cause soft rots in wood, and the soft rot is essentially a different kind of rot than the brown rot and the white rots that you see in terrestrial habitats, right? So, and they are very important for the invertebrates because when leaf or wood fall in the water, the bacteria are there, but they cannot access that. Fungi make hyphae, make enzymes, lignins, cellulases, amylases, break the substrate down into finer organic matter, which is then accessible by invertebrates and other insects in freshwater habitats. And then finally, that food can become fish food, right? So I like to say, if there are no fungi, there are no fish, at least in aquatic habitats, right? They play a very vital role in the decomposition of these organic matter. So we are also interested in looking for enzymes. We have a collaboration ongoing with the university in Toom, where we're interested in finding natural enzymes from these fungi that we can use for detergents in the future, hopefully. Here is a picture showing you how the leaf looks like when it decomposes. And like I said, these spores break down the organic matter, and then, of course, invertebrates eat it, and then that becomes the fish food as well. So they're very important. This is a picture showing you the view of wood under the microscope, right? You see these are different species of fungi. It's really hard to see, but these are different species. This piece of wood is from one of my collaborators from Australia, Sally Fryer. So it's tropical wood, so it's very rich, has all kinds of fungi. Here you can see nematodes and some insects and stuff. So why are we studying them? Because this wood is a long-lasting substrate. There is fungal competition with bacteria, competition with other eukaryotic organisms. And therefore, my professor, Dr. Carol Scherer and Jim Glore were the first folks to start looking into the chemistry of these organisms and studying them for their bioactive compounds. At Greensboro, we are also interested in chemical mycology and drug discovery with Nick. I'm interested in the taxonomy and systematics, and Nick Overlees is interested in the chemistry. North Carolina provides a perfect environment because of these unique environments from the mountains to the coast. When you straddle across this region, you find all kinds of different diverse habitats from which you can collect these organisms. Here's just a map of showing you some of the species. So far, we have approximately 150 or so in culture that we are studying. I also have a grant from the Indianapolis Zoo for fungal conservation, and with Moni Bohm, who is a curator of the aquatic conservation area, I'm studying these fungi for the taxonomy, and now once we get them in culture, we're looking at the chemistry as well. So just to quickly show you the chemical methods, we have mycology and then chemistry. Get the substrates, look at the morphology, get it in culture, sequence them, grow them in large scale. Once the solid state cultures are available, the chemists work on organic extractions, purifying it, fractionation, identifying the molecules, right? That's our procedure. So what have we discovered? A lot of new fungal taxonomy, because these fungi are so poorly studied, and also they're a rich source of chemistry. And today I'm going to talk about three examples specifically. One is a study of a grad student in the lab, and then one is an older study which we did with Pepe, who's in the audience when he was a postdoc at UNCG. And then another example that I actually worked with Manuel while he was at UNCG. Both of these guys are now professors here at the Institute of Chemistry. So I thought I'd highlight those studies. So in terms of mycology, we're finding a lot of new species, new genera. We have also found a new order from North Carolina. I mean, you would think that North Carolina is not that diverse, but it's actually very diverse. In our own backyard, if I go and collect, I find all kinds of species. So this is a very poorly studied group in the aquatic habitats, and obviously a lot of potential to discover new things. This was a new genus that I described recently from Indiana. I apologize for my screen. You can see the morphology here, and we called it the Indiana sooty sport fungus because it's a dark structure here. It becomes dark as it matures. So there's a lot of new diversity. So what are we doing? We are basically screening these fungi for different targets with different collaborators. I'm going to talk about Thea Cameron's work a little bit. She's a PhD student in our laboratory currently. And we're looking at antimicrobials, both try to find quorum sensing molecules, and I'll talk a little bit about that. Anti-malaria assays to find a lead against the Plasmodium parasite, as well as cytotoxicity studies with Dr. Joanna Burdett at UIC in Illinois. Obviously, all of this done under the umbrella of Nick's lab. So this is a story where we got these cultures from North Carolina. This one was right behind the music school where my wife studies music. And these two fungi were very interesting because I had never seen them before. When we sequenced them, we found that this is a rough tree, phylogeny with ITS and tubulin. You can see that they come out by themselves, and both of them are probably new to science. We need to describe them. There are about 30 so species in this genus Marinia. They are not quite very much aquatic. They are terrestrial and aquatic both. And we're trying to figure out the taxonomy of these strains right now. So what we did, Thea grew this up on rice, and you can see we found a really interesting compound here. We're working on the structure. This is the original and the regrowth. We want to make sure that our culture is always reliable and produces the compounds we need. And then this is 610 chromatogram compound A. And then it also found some lipopeptides here. These have been described in the past by Gerald Bill's group, and they've sequenced the genome, found the biosynthetic gene cluster for this particular compound, and it was also from the same genus Marinia. So it's interesting that taxonomically, this one also makes the compound. So compound A, we are still working on the structure. Like I said, it's got a long aliphatic chain, and we are interested in screening them for MRSA. MRSA is a big issue. There is a lot of cost involved, higher hospital stays, longer hospital stays, a lot of dollars spent, and high mortality, right? So we need some treatment modalities for MRSA. So especially when it forms the biofilm and when it makes these toxins that cause dermonecrosis. Ingrid, who was Pepe's student actually, is a postdoc at UNCG, and she tested the compound against MRSA, and we found that it was really active, and this is vancomycin, so you can see it's a good lead. So that's where we are right now with this. But because the MRSA is resistant against antibiotics, we are trying to see if we can use other ways to curb this problem. So this is just a sort of shot of the outside and the inside wall of MRSA, and essentially these accessory gene regulators control toxin production. I'm not going to get into the details, but basically this auto-inducing peptide comes out via the AGRB, gets cyclized, a quorum is formed. That means bacteria know, okay, we are good to go, let's attack the host. Then the AGRC gets activated, which in turn activates AGRA. When that happens, the problem really starts because this RNA3 transcript turns on promoters, which induce toxin production. And then this is the feedback loop, and all the other proteins are also activated. So can we find inhibitors for this issue here? So we've been working on that. We published a paper some time back. We found some prenatal diresorcinols from this strain collected at a state park. Nick mentioned we have permits to collect at state parks throughout North Carolina, and we are screening, we are going out and collecting submerged wood from these habitats, bringing them back to the lab and investigating them for diversity of fungi and chemistry. So we got this culture here. You can see I'm growing it on wood because it was growing better when you put wood in the media. This is the phylogeny of the fungus. It comes out in a group of fungi that are actually aquatic. Vibrissae is an aquatic genus. Loromyces is an aquatic genus. We were not able to identify to genus and species level because we only had hyphae to work with. There was no real morphology there. That's a problem. So we just left it at that. Solid state fermentation on rice was done. We grow them in the laboratory in large scales to get good amount of material. We grow them in Erlenmeyer flasks. Also, we grow them on mycobags. These are 100-gram mycobags, which enable for us to make more material. And we found 3 new compounds, prenalated resorcinols. We call them leoshiomycines A and C. We found that leoshiomycin A was active as an inhibitor of AGRA. What you can see here is the compound on the x-axis and the relative quantification of different, this is the RNA transcript, this is the phenol modulins and alpha hemolysins, all toxins that should not be turned on. Once they're turned on, you have dermonecrosis and you have a really bad wound. So you can see the addition of compound decreases their turning on, which is a good thing, and then your body's immune system can actually take care of the infection. So basically you can see this was inhibiting the AGRA in the assay. This is a docking model to show you that it binds to the AGRA, and you can see leoshiomycin A, this is the one in blue. This is more an optimized view showing the hydrogen bonds with the amino acids at the active site. And we have also found couple of other inhibitors through our work with fungi. This is omega hydroxyimodin. This was the first project I worked with Dr. Figueroa when he was in Greensboro. And then epicidin, which is another molecule that inhibits AGRA. So you can see fungi are actively making molecules that inhibit virulence in these MRSA strains. So these are good because you want to make sure that the MRSA doesn't get resistance to antibiotics. So this helps with resistance. Although in the literature now I read that even these proteins are slowly becoming resistant. But AGRA is a very good inhibitor. And if you can inhibit that, you're shutting down toxin production. So that's a good thing. The last story I want to mention was about guttates. I was working with this fungus in the laboratory, and I came across this fungus, which is a lachnum species. This is ITS phylogeny, and you can see these are all lachnum, and this is our strain G1177. And I was very interested in seeing what these were, because in the previous work we have shown that these guttates are actually chemical bombs. There's chemistry in them. They're not just droplets of water or dew. There is interesting chemistry in these guttates. So I went to Manuel, who was there at that time, and this is another picture from the internet. These two are the same fungi. You can see every time they're fruiting, they make these white water like guttates, droplets, exudates, right? So we were interested in seeing, I was interested in seeing what is this? Does it have chemistry or is it just water? So I went to Manuel and said, Manuel, can we do something with it? And Manuel said, yeah, let me take a pipette, get some of these guttates and shoot them in the UPLCMS. He did that, and you can see we found different peaks, and you can see the relative abundance. I mean, this is a lot of chemistry there. And this is the MSMS of that particular peak. Here's another one, MSMS of that particular one. And basically, in total, we found 5 from the guttates, and the retention time and the mass are here. So these 5 molecules were not in the dictionary of natural products. Manuel told me that I cannot find a hit for these. So these are probably new chemical entities that are still to be described. Unfortunately, we didn't have enough material to pursue the identification, structural elucidation of these. And then in the extract of the fungus, we found several other compounds that were known. So I think when you see guttates in your petri dishes and in your fungal cultures, I think they need to be explored. There might be interesting chemistry that the fungus is trying to say, don't come near me, maybe I'm going to kill you with my chemical bomb. We tested them against a suite of different antimicrobial targets, but unfortunately we could not get any hits. So that was kind of disappointing, but because we ran out of the guttates, we were not able to do further studies on this. But I have the culture, and I am hoping to see if I can get these to fruit in culture. And if the fruit in culture, perhaps on some wood, then I can collect the guttates and pursue and do more work on them. So, so basically, freshwater fungi are understudied source of novel secondary metabolites. And sometime back, Mario, myself, and Tamam wrote this paper. It was the first comprehensive review where we sort of outlined 199 chemical compounds out of the 280 reported from freshwater. Jim Glor's work was not all taken into consideration because there was already a review he'd written some time back. So if you want to learn more about freshwater fungi and their chemistry, how to collect them, how to identify them, this is a good review to look at. And finally, I would like to conclude by saying that, basic and applied research, you can do a lot. You can find promising leads that can become, new medicines for the future. I've thoroughly enjoyed working in a national products group. Previously, my work involved taxonomy and systematics and ecology, but it's really rewarding to see that these beautiful fungi also make beautiful chemistry that is worth exploring and studying. And finally, I would like to thank all the folks, including Pepe, Mario, Manuel. It's been a really enriching experience working with you guys, learning from you, and I hope we can collaborate in the future. And of course, Ingrid, who did some of the assays, I'm still working with her. My undergrads in the laboratory, Nick and Naja, and of course, some of the work that I showed was Tea's and Noemi Pagagins, who's now got a PhD. Finally, I would like to just have a plug here. There's a conference coming up in Asilomar, California, where I'm chairing a session with Dr. Ramon Batista, actually from UNAM as well. It is in March, and our own Dr. Figueroa is going to give a talk there on bioprospecting marine and freshwater fungi from Mexico. So hopefully that'll be a great session. And I'd like to thank you for your attention.
Thank you, Jose.
We have time for a couple of questions.
Yes.
Well, me first.
Mm-hmm.
When you're testing for your compounds that you're testing for antibiotic activity, once you find have no direct antibiotic activity by their own, do you discard them, or do you also try to find out if they can act as adjuvants for the antibiotics that are now resistant, MRSA, for example?
Yeah. We have not been working on that study as much. We don't discard them. We have them in the library. We are interested in if there is a way, a collaboration that we can work with and explore those. We'd definitely be interested in doing so.
Thanks.
Yeah.
Hi, I'm Dulce from Autonomous University of Baja California. I'm curious about the process for predicting the number of BGCs in fungi. Do you use any softwares? Because like internet and distance complicates that a lot.
So we don't do that per se. We collaborate with folks, and it's not very easy. It's extremely difficult because, especially with freshwater fungi, I don't think a single BGC has been predicted. When I was talking about predictions, I was talking about Aspergillus and Penicillium, which are very well studied. So yeah, we are working with a collaborator at Vanderbilt, and they are interested in doing so. So they sequence the genomes, and then we do the chemistry of our other fungal strains, and we work with them to see if we can predict and how we can predict. And we have a paper I can talk to you later. You can look at which recently came out, in where they show how this is done. What are the pros and cons of doing so? And one of the things they mention is that it's challenging with fungi.
Well, we're a little behind. Okay, last question. Quick question.
Yes.
Well, actually, I have two questions.
Yes.
The first one is related with this chemical entity that inhibits the AGRA.
Yeah.
I understand that is related with the quorum sensing and maybe is related with the biofilm formation capability of the Streptococcus strain. Okay, so that means that it inhibits the biofilm formation, then most of the pathogenicity of these strains are not expressed in the infection?
Yes, we did that only in vitro with RNA-seq essentially. But yeah, that is the premise in that it inhibits the AGRA, which is if turned off, then the RNA transcript and all the toxins are kind of inhibited. So
okay.
Technically speaking, you would not have an infection. We have not done work with this particular compound in mice models or other models where, in vivo models, where we can see how that works. Does it answer your question?
Yes, thank you.
Yeah.
So my second question, the phylogenetic analysis you are showing, you are using a collection of genes, a single gene,
yeah,
28 years, or the whole genome?
No, I'm not using whole genomes. I'm using single genes. I'm using ITS, the internal transcribe spacer region for fungi, which is a barcoding marker. I'm also using single copy protein coding genes to add to support of the clades. So sometimes single, but sometimes 2 to 3 genes, depending on the group that I'm studying, because it's very important to look at what data is out there to compare it to. And so I look at the literature and then sequence the genes based on what's available to compare it to. And so sometimes it can be 1 gene, sometimes 2 or 3. But whole genome is really hard because for freshwater fungi especially, only less than 10 genomes have been sequenced. It's still very early in that group. So yeah.
Thank you very much. Thank you.
También tiene... Maybe just plug it directly to the...
Cuando la quito, sale la silla. Ya no me voy a esperar. Sí, el HDMI. Pero sí entró, nada más que creo que toma más tiempo. Cuando la desconecto, de la... Ahí va. Creo que sí la corto tanto. Bájale la resolución. Y la resolución de salida. No va a quedar tantito. Y si le ponemos a ajustar, creo que luego saca las cosas de... Ah, ya quedó. ¿Sí?
Ah, okay. Sorry for that delay.
Bueno, nuestro siguiente ponente es la doctora Tamara Rosenbaum, quien es bióloga de aquí de nuestra universidad y después hizo su doctorado también en la UNAM trabajando con fisiología de las células beta pancreáticas y cómo su función es regulada por diferentes canales de calcio iónicos. Después realizó una estancia postdoctoral en Long Island, en la Universidad Estatal de Nueva York, en Stony Brook, y se trasladó después a la Universidad de Washington, en Seattle, donde estudió las relaciones estructura-función de canales iónicos regulados por nucleótidos cíclicos. Posteriormente se interesó en los canales TRP, que es de los que creo que más nos va a hablar, y estableció una línea de trabajo en el laboratorio de Sharona Gordon. Tras algunos años allá, fue contratada en el 2004 para iniciar su propio laboratorio en el Instituto de Fisiología Celular y actualmente es profesora titular C. Como les mencionaba, sus labores actuales son principalmente en el estudio de los canales TRP y cómo los componentes estructurales regulan su actividad y sus funciones en el dolor, la comezón y otras funciones fisiológicas. Welcome, Tamara. Thank you very much.
Thank you for your invitation. Can you hear me there? Okay. I want to thank you for inviting me to this forum of collaboration between two nations. I am especially thankful to Dr. Mata and Dr. Figueroa for considering me for this talk. So I'm going to be talking to you about the work that we have done at the Institute for Cellular Physiology of UNAM in my lab, working around the discovery of natural compounds that have roles on pain and some of the lessons that we have learned at the molecular level. So I want to start my talk by telling you that philosophers have often looked for defining features of humans like language, rationality, culture, and so on, and I'd stick with this. Man is the only animal that likes Tabasco sauce. So I hope you're enjoying Mexican food, and you probably have a sense of what I'm going to be talking about today. So I want to remind you that the ability to sense pain allows organisms to survive. For example, this little girl is going to put her hand on a hot frying pan, and fortunately she has sensory neurons that have specialized receptors that detect this change in temperature and send a signal to an interneuron, which in turn activates a motor neuron and then goes to the muscle and tells the muscle to move so that this girl can get away from danger. So in order for this to happen, we need to have very fast, very effective activity of neurons, which consists in the traveling or in the generation of electrical signals. But how does this happen? Well, we know that membranes are hydrophobic structures, and they do not allow for the passage of charged molecules such as ions, which we need to generate the electrical signals. But nature has solved this problem by inserting certain types of molecules which are called ion channels and which have in the middle of their structure a hydrophilic region which is called the ion conduction pore or pathway, which allows for the passage of ions, of charged molecules, and which can generate electrical signals. How do these molecules look? Well, many of them, such as this one, which is a potassium channel, look like teepees or Indian American houses which are inverted. And in the middle, they have this structure that I told you about, which is a pore, which allows for the passage of water and ions. And if you were an ion and you had to travel from one side of the membrane to the other, you would have to follow this path. Okay, so I'm going to center today, there are several types of ion channels, but I'm going to be talking about transient receptor potential channels or TRP channels. These channels are very well represented in all types of organisms, and they play several roles in physiology, which include phototransduction, osmotic regulation, detection of mechanical stimuli, calcium homeostasis, and taste. I will focus today on three other roles, which are thermosensation, pain, and itch, which is carried out by sensory neurons. So TRP channels are not only expressed in neurons, but they're expressed in several types of other tissues where they play different roles, and they contribute to normal physiology, but are also extremely important in disease. And there are several diseases which are, for example, hereditary diseases in which there are mutations in these channels that produce terrible sicknesses like skeletal muscle dysplasia, etc. So there is a very close relationship between the structure and the function of ion channels like in any other proteins. And channels are activated by specific stimuli, like there are channels that are activated by voltage, some are activated by ligands, etc. Among TRP channels, we find those that we call thermotrips, which are activated by different temperatures that range from cold temperatures that produce pain, there is nociception, intermediate temperatures, but also very hot temperatures that also produce pain and damage. So I just told you that these channels are activated by specific stimuli. And the reason why trip channels are my favorite channels is because they break the rules. And not everything is the way it seems. So apart from sensing different types of, different ranges of temperature, they also sense other types of stimuli, such as compounds that can be found in plants and that can cause pungent sensations or nociception. So today I'm going to basically be focusing on one of these channels, which is the TRPV1 channel, the vanilloid-1 channel, which was cloned for the first time in mammalians by the David Julius lab in the University of California, San Francisco in 1997, and then it was shown to be a receptor for capsaicin, which is the pungent compound of capsicum genus of plants. These channels are tetramers. I'm going to show you this a little bit further in my talk. They are non-selective cation channels. They respond to capsaicin, but they also respond to many other cues, such as basic intracellular pH, acid extracellular pH, and heat, nociceptive heat. And they are tetramers. Here I'm showing you the basic structure in a cartoon. The basic structure of these ion channels consists of 4 segments or 4 subunits that are identical, that have an intracellular N and C termini, and that have from 6 transmembrane domains, where S6 forms the pore or the ion conduction pathway. I'm just showing 2 subunits here for purposes of clarity. But if we look at the whole tetramer, you can see here in different colors what it would look like in a structure, which was also obtained by the Julius lab in 2013. And here I'm showing you the 4 subunits of this ion channel, and it's going to show right now that there are different agonists here bound to this ion channel. This is a toxin from a spider, and this is capsaicin here in the transmembranal domain. And here in the center is the pore or the ion conduction pathway. So I'm going to be talking very briefly about 3 stories from my lab, which most of them were serendipity based. And I'm going to, in order for you to understand how we obtain our data and what type of experiments we do, and also because we have students here, I want to tell you a little bit, very briefly, about the techniques that we use. We basically use a technique called the patch clamp technique, which consists of pulling glass pipettes and then forming a high resistance seal, a giga seal, between the tip of the pipette and the membrane of the cell. And the way this works is that we touch a cell, which can be a native expression system like a DRG neuron, or a heterologous expression system like HEC cells, and we form this high resistance seal. And what we can do with this is, with very sophisticated equipment, is that we can measure the activity of the channel. Here it was closed, now there's a Q, it opens, and it lets ions pass through the ion channel. Specifically, we use a technique that is called the excised membrane patch, which allows us to separate the membrane that's expressing these proteins, which can be wild type or mutated ion channels, and then separate it from the very complicated processes that are happening in the cell. So the first story started with us trying to understand what the structure of these ion channels looked like. I already showed you a very fancy structure from the Julius lab, but this did not exist when I opened my lab. So one of the techniques that many of electrophysiologists use to study function and structure relationships is called SCAM, or cysteine, or substituted cysteine accessibility method, which is based in exploiting the presence of cysteines in certain proteins. For example, TRPV1 has 18 cysteines, and using methyl sulfonate-based cysteine modifiers, which, cysteines are amino acids that are highly reactive, and MTS regions are specific for reacting to this with these residues. And what we measure is, here you're looking at this upward deflection that reflects the opening of hundreds of ion channels. So current passing through hundreds of ion channels in the presence of capsaicin. If we wash the capsaicin off and we add MTSEA, which is one of these MTS modifiers, we find that currents are activated, which is something that we did not expect to find with the wild type ion channel. So we went on to substitute all of the cysteines in this ion and create a TRPV1 cysteineless ion channel, which is no longer responding to MTSEA. So at that point, we were focusing on trying to find something called the activation gate of the ion channel, which is a region in the pore that allows for the passage of ion channels in a controlled manner. But I wondered whether there was something that was in nature that could actually interact with this ion channel, because these ion channels are activated by capsaicin, right? So we looked at extracts from onion and garlic, and we found that these extracts could activate currents, shown here in blue, from TRPV1. And then we used our TRPV1 cysteinless ion channel, and what we could see is that no longer these ion channels responded to these extracts, which meant that there was a cysteine, at least one, that was reacting with compounds found in onion and garlic. And so we went on, this is a long story, but I'm going to make it really short, and we found that the substitution of one single cysteine, which is this cysteine in the amino terminus of the ion channel, to an alanine produces channels that do not respond to the extracts anymore. And if we take the cysteineless ion channel and insert this 157 cysteine residue, we attain the activation by these compounds. So what are the compounds that we can find in these plants? Well, there are many of them, but we decided to test one that was a common suspect, which is allicin. And what we found that it was in fact allicin that was activating the channel, in the channel that does not have this sufficient but necessary cysteine to activate the channel with allicin. There is no activation, but if we insert it, then there is activation of the channel again. And of course, there is a physiological relevance for this activation of the ion channels, which is that when we inject mice in the paws with this compound, allicin, we can evaluate if there's pain. And the way we evaluate it is measuring the time that they spend licking the affected paw and compare it to the vehicle. So as you can see, TRPV1 wild-type animals display this pain behavior, and if we mutate or if we use a mouse that is a knockout for TRPV1, this response is decreased but not eliminated, because there is another channel, which is TRPA1, that responds to allicin also. And so here's the cysteine. It is located in the amino terminus, and the signal that allicin binding to this cysteine conveys goes all the way to the pore and opens the ion channel and produces pain. Okay, so the next study that we performed was started by trying to determine whether there were endogenous compounds that could activate this ion channel and that were related to pain. So for 30 years, it had been shown that lysophosphatidic acid, which is a glycerophospholipid with 18 carbons with 1 unsaturation, was binding to GPCR coupled proteins, or G protein coupled receptors, and producing chronic pain. But we wonder whether it could produce acute pain, and whether this acute pain was produced through interactions with TRPV1. So we started by evaluating the same assay that I showed you with mice. We injected LPA, we saw that there was pain, and then with the knockout animals, we saw that pain produced by LPA was greatly decreased. And as we looked at the mechanisms that could be producing this pain, we came across something that was very hard to defend, because for 30 years it had been thought that LPA only functioned through GPCRs, which was that it was actually binding to the channel directly. And we looked at the structure, and we found that there was this residue in the C terminus of the ion channel in a region called the TRP box, which is a sequence of amino acids that is well conserved in several TRP ion channels, and which is known to be important for the gating of the channel, meaning opening and closing of the ion channel. So we mutated this lysine, which is positively charged, to a neutral charge, and then reverted the charge, and assumed that the phosphate in LPA could interact with this residue. And as you can see, when we got these mutants, the activity of the channel was greatly reduced in response to LPA, showing that this region is very important for interaction of this glycerol phospholipid with TRPV1, and which can bind, it can be bound to the membrane, and with a phosphate group hanging, could be interacting with the C terminus and then promoting this conformational change, which opens the pore and allows the ion channel to promote a signal that causes pain. Now, my message to students is never underestimate serendipity. So when we had this work published, we were now wondering what are the characteristics of the fatty acids that are needed or required to be able to activate the ion channel. So we went through a big list of compounds that you cannot see here, but we found that at least we needed a length of 18 carbon tail, a phosphate head group, and one unsaturation. But as we were doing this these experiments, we came across this other molecule, which is called oleic acid, and which is basically LPA, but without the phosphate group. So one of the students was performing experiments, and she noticed that when she activated the currents of TRPV1, both at positive or depolarizing voltages or hyperpolarizing voltages, currents were activated, and then if she washed capsaicin and added oleic acid, then we could not activate the channel anymore with capsaicin. So this suggested that this compound was acting as an inhibitor of the channel. So instead of activating the channel, now this compound that is very similar to LPA is inhibiting the ion channel. And as you can see, this inhibition, when we activate the channel with capsaicin and then we wash it, and we add capsaicin together with oleic acid, the channel tries to activate, but then it's inhibited, and we can recover it partially. We can recover activity partially after a while of exposing the ion channel to capsaicin. It inhibits the channel with a dose response, in a dose response dependent fashion, with an IC50 of around 2.2 micromolar, which is not bad for these ion channels because these ion channels respond to compounds in the order of micromolar. And it also inhibits pain produced by capsaicin and by LPA. What's interesting is that it not only inhibits the activity of TRPV1, which is a polymodal ion channel, in response to capsaicin or LPA, but also to temperature. So if we activate the channel with temperature, we have this response, and when we add oleic acid, this response is basically dead. So it's inhibiting the response of the ion channel to temperature too, to pH, to extracellular acid pH, which is a condition that you can have when you have certain diseases of the heart and you have ischemia. And also it inhibits the activity of something that we discovered in this paper, is an inducer of itch, which is called cyclic phosphatidic acid. And hence we looked at whether it could inhibit itch in mice, and we can see that it does inhibit itch in mice. And this is really not a surprise because there are many creams and soaps that are used that have olive oil where this molecule is present, and that have been used for itch conditions. So how does it inhibit the channel? Well, we've done this assay for many of the studies that that were shown today, but I'm going to just show this one today, which is by decreasing something that is called the open probability. You can look at several hundreds of and thousands of ion channels in a patch, in a membrane patch, but you can also look at a single molecule in real time. This is the beauty of patch clamp. So if you look at one single ion channel, you can see how it opens and it closes very little in the presence of a saturating concentration of the agonist, so the open probability is high. There is no OA here, but when we add OA and we let it stay for 40 seconds, you can see how the open probability decreases with time. And this means that this compound is stabilizing the closed state of the ion channel. Okay, so how does it act? Well, capsaicin, as I told you, binds to a transmembranal region. It interacts basically with three different residues, which are a tyrosine 511, a serine, and a threonine. And after many, many experiments, we found out that the way that this compound was acting was in a very surprising manner again, through an interaction with this region. And the way that we ended up discovering this, because I'm not showing you all the experiments we did, was because we tested the chicken TRPV1. And some of you know that if you give a parrot chili pepper, it will eat it. And the reason for that is because it's not getting the hot, pungent sensation that you get from chili pepper in mammals. And that's because there is one residue, only one residue that's different, that does not allow for capsaicin to be stabilized. So when we tested mutants, a double mutant of this site, we saw that after OA, the inhibition was much decreased in comparison to the wild type ion channel. And if we test The triple mutant, you can see that OA simply does not inhibit the ion channel. And if we turn a chicken TRPV1 channel into a rat TRPV1 channel just by mutating one site, we can recover the effects of oleic acid. And so here's a model of how it binds. It interacts with these 3 residues, and it gets into this binding model is the typical binding model of a key and a lock, where it specifically binds this little pocket called the vanilloid binding site. And this is one of the problems, the most interesting problems in biophysics, is how does something that binds to a region can activate while another one can inhibit. So this is something that we're trying to understand now. Now, my last few slides are about one of my favorite drinks. And so there is a story behind why we started testing this compound. This compound, alpha-humulene, which is a sesquiterpene found in cannabis and in hops, which give this hoppy taste to beer, is a compound that has been used for thousands of years in Chinese traditional medicine, and that is continued to be used now in creams to produce some pain relief. And the reason why we started testing this is well, I don't want to. Okay, so we went to a meeting in Chile, and I had my child with me because I'm a working mother, and she was outside of the meeting talking to people with her father, and she petted a dog, and this dog bit her in the face. So she had a lot of pain, and one of the persons here in Chile that was our friend, had this cream, and she gave her that, and it actually worked. So many years after that, I remembered, and I looked at the compound, and it was this one. And so we began to test whether alpha-humulene could produce inhibition of pain through TRPV1, and we got the compound and started by injecting the mice with alpha-humulene and capsaicin. As you can see, the response is decreased again. Alpha-humulene by itself does not produce any effects. And if you go to the DRG neurons, which are the native expression system, you can see that there's a correlate for this physiological result, which is that when you activate currents with capsaicin and then you add alpha-humulene, DRG neurons do not activate in the presence of capsaicin anymore. Okay, inhibition of the channel is dose dependent, and it's more efficient when the channel is not in its most open probability, meaning you go from 0 to 1, it's around 0.5. So the channel, it prefers the channel to be closed, which is already pointing out to, again, a mechanism that induces a stabilization of the closed state. So does AH modulate the activity of other ion channels involved in pain? This is a typical question of reviewer number 2. And of course, there are many ion channels that are involved in pain. TRPV1 is not the cure for everything. It's just one of the effectors. So TRPV4 is another channel that is involved in pain, which is important. We tested on this channel, and we saw that a very high concentration of this compound could inhibit the currents of TRPV4. But if we look closely at the dose response, you can see that the IC50 is shifted to the right, meaning that it's not so sensitive to this compound. Now, if we look at another TRPV5 channel, we can see that it just simply does not have any effects on this ion channel, meaning that there is some kind of specificity of this compound. And then, of course, there are sodium channels. Sodium channels are extremely important in generation of pain signals. So we looked at the common suspects, which is sodium channel, voltage gate is sodium channel 1.7, and we saw that alpha-humulene did not have any effects on the currents of this type of ion channel. And also to eliminate the contribution of any other sodium channel that we could find in DRG neurons that express TRPV1, we just cultured DRG neurons, applied alpha-humulene, and we could see that activation of sodium currents remains, which means that it's not affecting sodium currents. So we then wanted to find out where this molecule was binding. We were having a hard time finding the region where it was acting. So we got into a collaboration with Alexander Sobolevsky at the University of Columbia in the United States. And he is the genius of cryo-EM. And as you can see, here's the structure of the ion channel with alpha-humulene here shown in red in a transmembranal region. Here's a another view of this interaction. And if we do a close-up, you can see that this region involves residues that are located in the S2 and S3 transmembrane segments, and interacts with certain types of residues. And this is a site which is completely new because nobody had previously shown that this region of the ion channel, which is all the way in the beginning of the structure, has anything to do with the gating of TRPV1. So we looked at the map that they generated closely, and we did, in collaboration with the Vergara lab, a series of MD simulations, and found that there is an increased contact frequency with alpha-humulin in these residues. And so in order to show this physiologically, we mutated the residues individually, and we saw that those responses to alpha-humulin were shifted to the right, and the channels became less effectively inhibited by this compound. Unfortunately, we could not mutate all of them together because the channels did not functionally express, but this together with the structure suggests that this is the binding method or binding site for alpha-humulin. So with this, I want to finish my talk. I want to just show you some of the work that we have done with natural compounds. And I want to thank all my collaborators and my funding agencies from UNAM and CONACYT. And I ask you to please support basic science. Thank you.
Thank you very much, Tamara. Tenemos tiempo para un par de preguntas.
Thank you, Doctora, for this wonderful talk and the wonderful material. It's very interesting the way that you presented. I have a easy question. It's about sometimes people perceive that someone can have more pain or can be so susceptible for more pain.
Mm-hmm.
Is it true that we could have more receptors, or is it something that is really physiological?
No, it hasn't been proven that we have more receptors, or there are populations that have more receptors. But what can be, for starters, pain is a subjective sensation. So if you're in pain today and tomorrow you're depressed, you'll have more pain tomorrow with the same stimulus. It also depends on your state of health. If you have inflammation, these are polymodal receptors, and that's why they're so complicated, but then so beautiful that you can, what you find in these ion channels, you can project to the understanding of other proteins. So one typical question that relates to this is, why are Mexicans resistant to chili pepper? This is not the case. A child, you don't take a Mexican child and give it a hot sauce and it will not react, or it will not react. Of course it will react. You get accustomed. There is a cultural component of eating chili peppers, and you go out with friends and they tell you to eat a chili pepper, and so that's how you learn how to eat it. We don't have less receptors. We just get used to it, like many other cultures.
Thank you very much for your talk. Which is the best ion channel for prove neuropathic pain?
That's a good question. TRPV1 is involved in a lot of processes that involve neuropathic pain, including diabetic neuropathy, Dr. Mata. But there are other ion channels that are also involved in these processes. So there are so many things that go on when you have inflammation. So trypin-2, for example, is another type of ion channel which is associated to chronic pain. One thing is nociceptive pain, which is chili peppers or other things that go away in 1 or 2 weeks or immediately. And another thing is chronic pain, which just persists. Okay, things there are modulated in a different way, and this is why this is a health problem that has to be studied from different levels, because we just cannot solve why we have chronic pain. Trypin-2 is a channel that is, for example, present in white blood cells, so it has to do with inflammation, but there are a bunch of other ion channels that respond to prostaglandins like TRPV1, TRPV2, etc. I mean, there is several ion channels involved.
Go ahead. Okay, I really enjoyed your talk. I loved your slides. So I was really curious in the hops example. Do we have trip channels in our estomaca, or are we absorbing the alpha humulin and it's getting into our system somewhere else and then stimulating?
There are trip channels in some of our epithelium and endothelium, etc. It does cause irritation because of other properties, not TRPV1 necessarily. And if your question is why do we have, I'm trying to understand your question, why do we have heartburn, for example?
No.
No.
I love drinking beer.
Okay. No, so, yeah, I think that many diets, including beer, which have olive oil, have anti-inflammatory properties that have been proven. So it does get absorbed to a certain level. Yeah.
Hi, just a quick question. If you could give us a reflection on, because I was impressed a little bit about the slide where you show that TRP are like widespread in all the body, no? Do you think it could be a good drug target for like nociception or many other, like, because there are like wide activities control so many, and I don't know if you have any experience or through all the research you've done.
Well, I can answer your question with a very big problem in the area, which is if you target TRPV1, you're targeting an ion channel that responds to heat. And there's a lot of work where you inhibit TRPV1 and what you have is hypothermia. So this is the problem of finding something that will inhibit TRPV1 to produce, alleviation or inhibition of pain. There are things that you can use topically, like capsaicin itself. There are patches that have capsaicin, you put it on, it desensitizes the ion channel, and then there's no pain, or these creams. But as in terms of something chronic, I have hope that there will be, I think that, and there's agreement with David Julius, which is the Nobel Prize that discovered this ion channel, that there will be some natural compound that can be used. But at this point, our main problem is hypothermia.
Thank you.
Thank you very much for your talk. I really enjoyed especially the intro with all the graphics and everything. It's very immersive. My question is about the alpha humulin. Have you looked into the possibility of using as a ligand in a PROTAC, like a protein degrader, as some sort of medical solution for pain?
Alpha humulin as a protein degrader?
No, as part of it, like the protein binding ligand.
Oh.
So connecting it to another ligand, which can then link to the proteasome system. Do you think it's a possibility in this?
I suppose it is a possibility. You're thinking, I'm trying to understand because I can't hear you very well.
Oh, sorry.
But you want to know whether alpha humulin, a molecule that's similar to alpha humulin system, a binding system? Well, yes, of course. So many of the things that we have for medicine include finding out where they bind, molecules bind. Cardiac medicine for tachycardia, for arrhythmia, etc., are all ion channels, and understanding where they bind. So understanding how these different binding regions, like the BBD, the vinyl binding domain, which instead of opening the channel now closes it, is I think one of the things that we have to pursue to find something that will allow us to sustain. Alpha humulin, I think it's a very interesting molecule. It is the one that we have found that is that has the more affinity for the ion channel and inhibiting. So I want to kind of pursue that a little further.
Yeah, it's interesting that it has such a potent activity, even though it's such a small molecular weight compound.
Right, it's very small, and that's what we had trouble with
Yeah.
in the beginning.
And allosterically active.
Right.
Yeah.
Bueno, thank you, Tamara, again. Let's give her a clap. Bueno, tenemos, por favor, 15 minutos para receso, 15 para coffee break. Y los que no se han registrado o que no pasaron lista, allá afuera ahorita va a estar la lista.
En las diapositivas de las próximas.
Sí, por favor. Tiene que ver ¿Vas a conectar o traes?
Oh, me da igual, pero puedo conectar, sí. Pero traigo un USB chiquito.
¿Puedes conectar o la pasamos? Yo tengo la primera.
¿Es este? Ajá. Sí. Ah, súper bien.
Como vamos a poner un zoom para los que no van a venir.
Sí, claro.
Mejor no moverlo.
Sí, mejor. Así más rápido.
Pérez Ramírez, quien se graduó como químico bacteriólogo parasitólogo de nuestra universidad hermana, el Instituto Politécnico Nacional, donde también hizo una maestría en microbiología y un doctorado, y posteriormente realizó una estancia postdoctoral en el CDC en Atlanta, Estados Unidos, en el desarrollo de vacunas, y además una maestría en administración industrial por el IPADE. A lo largo de su carrera, el doctor Ramírez ha trabajado en diversos aspectos de la biotecnología, incluyendo la fijación biológica de nitrógeno, la caracterización molecular de cepas, la expresión de proteínas recombinantes y el desarrollo de procesos. Actualmente es el COO o director de operaciones de la empresa biofarmacéutica mexicana Probiomed, en la cual dirige más de 400 personas gestionando diversos proyectos que incluyen desarrollo y producción de vacunas, procesos de síntesis química, formulación de fármacos y producción de anticuerpos monoclonales. Además de su trabajo industrial, ha dirigido, ha sido gerente de 14 proyectos del CONACYT. Tiene numerosas citas, muchos artículos en revistas especializadas, más de 70, y también ha sido tutor de estudiantes de distintos niveles. Es miembro del Sistema Nacional de Investigadores, de la Academia Mexicana de Ciencias, y también es integrante de la Comisión Biotecnológica de la Farmacopea de los Estados Unidos Mexicanos. Doctor, es un honor tenerlo aquí. Muchas gracias por su participación.
Gracias. Well, thank you very much for the invitation, Patricia. It's a pleasure to be here with you today to share a little bit about the innovation and the science we do in the pharmaceutical industry in Mexico. Okay, I want to talk just a little bit about the company I work for. We are going to talk a little bit about biosimilars. Maybe a lot of you doesn't know the biosimilars, which is good, because if you know, it means that you have, or somebody related to you have some very bad diseases that I don't want to talk about. Then I'm going to talk about case studies that I want to share with you today. So we are a company that we do our own developments. That means that we use something that is called quality by design to develop a product that it's a candidate. You know, this product candidate basically is a recombinant protein that is already in the market, and which patent is about to expire, right? So that means that we can produce the same protein with the same quality, the same attributes, and for that we need to use apply science in order to get a commercial product that needs to pass through all these steps during the research and development. One important part is the patent analysis, no, to guarantee freedom to operation. It means that we don't have any issue when we get out to the market with this biosimilar. And the most important thing is the product development and the development of the actual process that is going to produce this recombinant protein. So we are a company that we consider ourselves a knowledge generating company. We have by the time 45 research articles. Well, maybe it's not that much, no, but for a private company that depends mainly in the industrial secret, actually it's a big achievement. And we have already 3 patents granted. The history of our company: we were actually the first company that launched a biosimilar, not just in Mexico, but I want to say that maybe in the world. In 1996, we were starting the production of a recombinant protein that is out of the market right away, because there were some new products that substitute the treatment. The name in Mexico was Gramal, and the active compound was GEMCSF, which means granulocyte monocyte colony stimulation factor. And then we start working with interferons, the interferon alpha 2B, which is actually a product that is also declining his use because it was developed for the treatment of hepatitis C. However, now we have much better treatments for hepatitis C, so the molecule was not in use anymore. However, recently, a couple of years ago, some A-care workers discovered that interferon alpha 2B could be useful in the treatment of cancer of the eye. So this product is getting now a new opportunity in the market, and we are actually selling it. It's just a bit, but we start producing this molecule again. Then we produce our joyetin, which is human erythropoietin, and actually this is our gold brand. So 90% of the EPO that is sold in Mexico is produced by us. So we are the main producer of erythropoietin here in Mexico. We are the only ones that produce a recombinant vaccine against the hepatitis B. And we also launch some other products later on. This one, Filatil, which is the granulocyte colony stimulation factor, was the product that actually replaced the use of the GEMCSF, at least in Mexico. We produce also some interferon beta, for the treatment of multiple sclerosis, which is a really bad disease. This one was also for that. It's interferon beta 1A. And in the beginning of 2010, we launched our first monoclonal antibody, which is the biosimilar of rituximab. And then we also launched another, it actually is a fusion protein, which is Tanercept, under the name of Infinitum. So I'm sure that all of you have gone to the pharmacy and you ask for a drug product that you got a receipt, and then when you reach the counter, you ask, hey, do you have this product? And maybe the counter will say, hey, we have also our own brand for the pharmacy, and we have the original one, and we also have a different brand for a different company. Okay, and which is the price? Well, the original one costs 1,000 pesos. The brand of the other lab costs 500, and the brand for all pharmacy, because now you go to the pharmacy and the big chains, they have their own brands of products, and this one costs 250. Okay, which one did you choose? Honestly, which one did you choose? The cheaper one, right? So, or maybe it depends how much money do you have in your wallet, right? Because if you have enough money, you say stuff like, oh, the most important is my health, so I'm going to invest 1,000 pesos. But if you don't have the money, say, okay, give me the 250, right? Or maybe there are cheaper ones, right? So the proteins, they have an inherent complexity, right? And some people say the bigger the protein, the harder to characterize or to control the production and stuff like that. But we know that actually it is not actually true, because the most important thing for the production of a protein is that how much or how many post-reductional modifications the molecule has, right? So, for example, somatropin, which is produced in E. coli, it doesn't have glycosylation, it doesn't have any post-reductional modification, It's kind of tricky to do the somatropin because it tends to clip, and then you need to control the conditions to avoid this clipping. However, it's a pretty simple molecule, right? Then we have a monoclonal antibody. The monoclonal antibody are big entities, big molecules. However, the post-traductional modifications, they're just a few. So they have 2 glycosylation sites, which are actually very important because these glycosylation sites controls the effector activities that the monoclonal antibody has. It means the activity, the capacity of the monoclonal antibody to recruit immune cells from the immune systems, not NK cells, macrophage, actually the ability to bind some plasma proteins to control the pharmacokinetics. So it's actually very important, but it's in terms of chemistry, maybe it's not that complex. However, erythropoietin, it has 3 different glycosylation sites, and they need to be charged. It means you need to achieve a full glycosylation in order that this protein could fulfill the pharmacokinetics in the patient. So it's a little bit harder to control the glycosylation profile of erythropoietin. And this is just a curious thing, the heparin, which is actually a biological product that is extracted from the intestine of pigs, or porks, is actually, here we say, well, this is a pretty simple molecule. However, there is a range of molecular weights that could confirm the heparin. So the purification of a single entity, it's quite not. I mean, I don't want to say that it's simple because it's not, but it's easier that the purification of a collection of multiple molecules, right? To keep the activity that you need to get with them. No, the more variety of the molecules, you have chances to purify something different in the process. So there is some paradigms, no? Right? They say again some paradigms against the conventional and related with the biosimilars. So the innovator says different process means a different product, right? If you use a different process, then you are going to get a different protein. Different manufacture, different drug substance, and they say that the process is the product. But we know that if you know the product quality attributes, you could use quality by design to get the right protein using a novelty process, a control process that gives you a defined product, and that we know that the product defines the process. Something that is interesting is that most of these products were developed at least 20 years ago. Right? So maybe 30 or 40 years ago. So if this is true, different process, different product, that means that we would have to use the technology that was developed so long time ago, but today we have much better chromatography resins, different chemistry in the filters. We have now plastics that are not detaching different toxics in the product. So the advance of the science is in this size, in this part. So also we have in the chemical faculty, so there is a lot of QFB here, and in Mexico, QFB is the career that allows the people to be sanitary responsive in the production of a pharmaceutical company. So you need to know that the process to get a production permit or sales permit for a product, it's for a conventional molecule or innovator molecule, is based on basically the clinic, right? Because this product is new, you need to demonstrate that there is no side effect, that there is actually effective, that there is no safety issues. So how you do the analytics and the preclinical, maybe it's not that important because it's the only molecule, right? But when you are working with a biosimilar, the thing needs to be the positive way, because the most important thing is to demonstrate that you have the same molecule, or at least a biosimilar molecule, right? So then in the preclinical, you need to demonstrate that the molecule has the activities that are actually related with the mechanism of action of the molecule. And then the clinic phase is just a confirmation that the molecule is working. So this is in the Mexican law. And for that, in our company, we have developed a lot of different techniques for the molecular characterization of the products we are talking about, including these functional assays. These functional assays that are actually done in the preclinical part or non-clinical part are related with the specific mechanism of action of these molecules. So actually now WHO, here's WHO, sorry, has a new guidance for the evaluation of similar biotherapeutics, and using scientific principles, the most important thing is that the demonstration of biosimilarity is sufficient to ensure product safety and efficacy. That means that they rely on extensive characterization of the product. So as I told you, we have a lot of different methodologies that assures the comparability of the molecules using these 4 levels of protein, right? Primary sequences, secondary sequences, tertiary sequences, and quaternary structure. And that means that if you have no difference in the chemical and in the physicochemical and functional characterization, then your clinical trial could be just a simple PK/PD. Well, it's not that simple. It costs like 20 million pesos to perform a clinical trial in this a good PK/PD. So, but if you have some differences in the physicochemical and functional characterization, then you need to extend your clinical trials, right? Because it is detected as a risk that something is not going to work very well. And actually, we have an example. This is a study that was published for the Planetra study for a biosimilar of infliximab that was developed in Korea. And what they found is that they found some differences in the affinity to NK cells in an activity that is called ADCC, what means activity dependent of cytotoxic cells, which is actually a relevant mechanism in inflammatory bowel disease. So what happened, right? In the preclinical level, they have to perform increasing ADCC using different cell lines of different origins, different effector cell source, because they found this difference. And the clinical level, they need to do a greater post-marketing follow-up. And actually in Canada, the health agency did not authorize the use of this molecule for the bowel disease, but just for the rheumatoid arthritis. So the difference in the preclinical comparability might increase the number of the studies as well as the number of patients. So that's why we rely on a lot in the characterization of our products. So saying this, I'm going to talk about this case study. We actually, in MAPS in 2018, there was a study that was patrocinated by actually Pfizer, and they they measured the variability, something that they call intended copies of etanercept, no? And they use several batches of 7 different products. I'm not going to talk about the other products, but I want to talk about the product we produce. So they take 3 different batches or infinitum, and they they use all these products, and infinitum is the product that is produced by ProBiomed. So I'm not going to talk about the results of the others, but I'm going to talk about the results they got with our product. So as you could see, they use different identity tests like peptide mapping, hydrophobic interaction, size exclusion, the N-linked oligosaccharide map, purity measured by host cell proteins, protein A, which are contaminants related to the process, the charge distribution, SDS page, sialic acid content, receptor binding, and the activity which is related to the mechanism of action, which is the inhibition of TNF-induced apoptosis. So, and they report that the N-linked oligosaccharide map quality, they report the same percentage in neutral as isolated, but they say the profile is not comparable. So actually this surprised us, because we have done this before, and we were pretty sure that the profile we got in our lab was pretty similar that the profile they report, or the one we measured when we got from the pharmacy their product. So we measured the profile, and we found that it was more or less the same. And then we have this publication that we were talking about, the clinical trial actually we do for the biocomparability, and we were also reporting that the characterization of our molecule. And what we found is that we have different batches of Embrel, and we found that not all of them share the same carbohydrate profile. So we report, no, we are similar to the Embrel batch A. Batch B and batch C they have some differences. And then the same authors from the fake paper send a letter to the editor and says, you know, we said that this Embrel batch A is not our product. So we don't recognize this as the identity of the Embrel we are using as reference in our studies. So we actually send a letter back. And they say, you know, we make, this is the batch we use to do the comparability in our studies, and this actually is a standard reference, the Embrel, that's what reported by Pfizer. As you could see, they are practically the same. So what's going on here is that with time, these companies have suffered some changes, and this was published in Nature by Sandoz, and they say, look, this is Embrel. No, this is rituximab, and this is Embrel. And they say, you see, during the pass of time, there has been a change in the variability of the glycan profile of Embrel without any changes in the variability of the product. So what happens here is that they were working with this standard, and when we did the development, we were working with this standard. So that means that we were similar to this one, but not to this one, right? And then they say, okay, we are going to check, they check the manufacturing history to check the contents, the consistency of product quality through major process revisions. So this is actually the history, how the products change during, as reported by them, 4 different major changes in the production process. And as you could see, there are actually some differences in the reverse, in the percent of reverse phase area, the content of wholesale proteins, which they start having high quantity of wholesale proteins, and now they control the level, and the binding activity to the TNF alpha, and the potency. So as you could see, there are some changes in the quality of the product. However, they say, well, while the affinity and the potency are the same, there is not going to be any problem. So, but we decide to go a little bit further and say, okay, they say that, and this is because in the paper they say that slightly difference in the glycan profile could have an unknown effect over the quality, over the safety and efficacy of the molecule. So we decided to check what actually it means, right? So we took the molecule and we start, cutting the glycosylation profile. We use glucanases in order to cut the different levels of the glycan profile. And what we found is that you actually need to strip the molecule in order to see a difference. And the most important thing is that the last thing that you could see is the difference in the potency, but interestingly, there is a effect in the stability of the molecule. So that means that these traditional methodologies that we use in the pharmacy as the stability study is actually an indicator that the molecule is okay, right? So it means that slightly changes in the glycosylation profile are actually not that important, at least for this molecule. However, a catastrophic change could be detected running some traditional stability tests. So my last subject is rituximab. Rituximab is a monoclonal antibody that is used for the treatment of lymphoma non-Hodgkin, which is a type of cancer. And we know that rituximab is photosensitive. So it means that you need to be careful with the light during the production and during the storage of the molecule. And then we start looking how sensitive it is and what physicochemical attributes are affected, which critical quality attribute is affected if we have avoided. So that what we do is that we exposure rituximab to different levels of light. Actually, it was sunlight for different time, and it worries us because when you got lymphoma non-Hodgkin, you need to go to the clinic, and then you are in some rooms. The doctors, the physicians go, put the rituximab, and then introduce rituximab to you through infusion. So it means that the rituximab is exposed to light for some time, right? So, and we believe that we need to control the time that rituximab is exposed to light. And because something that is very important is that actually there was a decrease in several quality attributes. Actually, we determined that the light is causing the amidation of the molecule, and in the most important CQI that is affected is the potency. So we decide, well, what is happening? So we start doing some informatical work, and we find that actually the amino acid that is directly responsible to the binding of the rituximab to the CD20, which is the marker in the molecule, it's deamidated, and that's why the potency is decreased when rituximab is exposed to light. So what we're going to do with this information? Well, we actually develop a new formulation that protects the molecule for photodegradation, and in the aim to look for a safer product, and we are now in the process of patenting this novel formulation. So the conclusion is that there are paradigms about biosimilars that must be combated with knowledge. So we believe that knowledge is actually very important for anything that you decide to do in science, and the physicochemical and functional comparability should be sufficient to ensure safety and efficacy of medicines, and the knowledge about molecules allows the development of new innovations. Thank you very much. Aquí hay otro. Paso el otro.
Hello, thanks. How are you doing in your last biomolecule into making it better to not making it photosensitive right now, if I could ask?
Well, we cannot change actually the molecule. If we change the molecule, then we are start doing something that is called biobetters. So it means that there is a next generation, and you need to demonstrate safety and efficacy since the beginning. So unfortunately in Mexico we cannot afford to pay an extensive clinical trial. Basically because the price of the medicines here in Mexico is actually very low. If you compare with USA, for example, there is no way that we can compare the prices of the biotech products in USA and in Mexico, especially the ones that we sell to the government. And but what we can do is to look for better ways to protect the molecule, which is that what we are doing. So we are trying to develop novel formulations to assure the stability and the efficacy of the molecule in this case.
Thank you very much, Dr. Perez, for your talk. I thought it was really interesting. I'm a clinician and I use biosimilars in my job, and we use rituximab a lot, especially when we're treating oncology patients.
Okay.
My question is with regards to the photosensitivity of the product. This is something that we've all come across when we're administering it. The question of the photosensitivity and the reduced potency of rituximab when we're administering it, because it's photosensitive like a lot of other products. We have a relatively simple workaround at the patient basis, which was we just cover it in a dark bag, an obscure plastic bag, to prevent it from being directly exposed to sunlight. Is that something that is a simpler workaround to creating a whole new patented project, or is that actually not useful from your understanding of the research?
I think that is useful. Actually, the thing is not to expose the molecule to the direct sunlight. We try, to check what's going on in the places where you receive the product, and we notice that sometimes you strip the vial and you put it there while you are preparing all the stuff you need. So maybe we need to avoid that. So we need to put some better ways to do it, to minimize the sunlight directly to the product. That could be one important thing.
So it's more about every step of the process rather than just at the clinical administration stage. Thank you very much.
And in ourselves, as soon as we got the patent granted, we are going to change the formula, and we are going to declare that this formula is safer because it's photo resistance to the sunlight. So in that way, you don't need to worry for that.
Thank you, Dr. Nestor.
To the other side.
Yeah, it's
because it's only like where everyone puts their title, I guess.
It's hard.
That one. Yeah.
You have to put it here in the bottom.
So anywhere else, I guess.
Ok, nuestro siguiente invitado es el doctor José Rivera Chávez, de aquí del Instituto de Química de la UNAM. El doctor Rivera es químico farmacéutico biólogo de la Facultad de Química y maestro y doctor en ciencias por el posgrado en Ciencias Químicas de la UNAM. Posterior a su graduación, realizó una estancia postdoctoral en la Universidad de Carolina del Norte con el doctor Nicholas Overlis, donde estuvo por allá algunos años, y regresó al Instituto de Química, en donde fundó el Laboratorio de Química de Productos Naturales, fúngicos, en donde estableció una línea de investigación que integra la química, la biología para el estudio de los compuestos bioactivos producidos por hongos, con aplicaciones potenciales en el tratamiento de enfermedades infecciosas y crónico-degenerativas. Tiene una vasta producción científica, también ha graduado numerosos estudiantes de diferentes niveles, y a nivel internacional es cofundador del consorcio Glacier, que fue financiado por la DAAD, una red que promueve la investigación sobre enfermedades infecciosas en México, Alemania y Cuba, y asimismo colabora en diversos consejos académicos y comités de alto nivel, tanto nacionales como internacionales. Thank you, Pepe, for accepting the invitation, and welcome.
Good morning, everyone. It is really a pleasure to be here today to share some of the results we have gathered over the last 8 years regarding the chemistry and biodynamic properties of secondary metabolites produced by Mexican fungi. Before I start, I would like to thank Dr. Mario Figueroa for the kind introduction, and also for the invitation to participate in this great symposium gathering natural product scientists from Germany, the United States, and also from Mexico. So, in the following minutes, I will try to summarize our work we have done in an ant-associated fungus through the talk titled Exploring the Chemical Space of Thalaromyces amestolchiae, Natural Products and Semisynthetic Opportunities for Drug Discovery. Well, this story began around 8 years ago when I was hired as a faculty member at the Institute of Chemistry at UNAM, where I established the Fungal Natural Products Lab and also implemented 2 major research projects: the application of multi-informative strategies for the integrated and chemical biological study of fungal microorganisms with emphasis on the characterization of their secondary metabolites, and also the design and execution of in vitro biological assays aimed at discovering enzymatic inhibitors and also compounds capable of suppressing the growth of resistant bacterial strains. But you may be asking why to study fungi or natural products from fungi and why now? Well, as Josefa mentioned in the previous talk, fungi are taxonomically and morphologically diverse, and also an understudied group of microorganisms that in fact hold a great potential for many biotech applications. These organisms are capable of degrading complex materials and transform them into useful molecules that offer cost-efficient solutions for many human problems. So, the metabolic and genomic plasticity of fungi make them useful for the efficient production of many enzymes, organic acids, nutraceuticals, and also novel natural products with impressive bioactivities. Some examples of them are, in fact, some antibiotics, cholesterol-lowering drugs, and immunosuppressants that have become drugs that have saved the life of many people. So traditionally, the study of natural products or the discovery of bioactive natural products is performed following a bioassay-guided isolation protocol. However, using this kind of methodology, drugs such as Taxol, ivermectin, artemisinin have been discovered. However, over the last 2 decades, the application of this strategy often results on rediscovering known molecules. To face this issue and taking advantage of modern analytical tools, we have now implemented a multi-informative approach that combines several layers of information. In our particular case, we integrate a metabolomic information, also taxonomy and bioactivity data, and integrate all this information in molecular networks that help us to prioritize the most promising fungi for the isolation of new bioactive compounds or interesting compounds with unknown bioactivities. The main health issue that we are working with in our lab is type 2 diabetes mellitus. This is by far the most prevalent chronic disease worldwide, with estimates with numbers that estimate between 589 million people to 800 million people living with this disease. Type 2 diabetes mellitus is closely related to insulin dysregulation. So molecular target that could regulate insulin and glucose levels in patients with type 2 diabetes mellitus is protein tyrosine phosphatase 1b or PTP1B. This enzyme is a negative regulator of insulin and leptin signaling, and its inhibition could lead to reduced glucose levels and also minimize the micro and macrovascular problems associated with type 2 diabetes mellitus. So based on this perspective, about 8 years ago, we started traveling in Mexico to collect some samples and establish a fungal collection. So we sampled many places, and trust me, it wasn't as funny as you think it was, because as Josefa mentioned before, also we have to face some alligators or crocodiles. We didn't return back to ask them what they were, right? So we sample plants, soil, sediments, ant hills, dung from different animals, and these collections were done in semi-desert soils called cloud forest, mesophilic forest, oak and pine forest, wetlands, and ant hills, and also we collected some lichens. Then we bring these samples to the lab, and applying several microbiological processes, we were able to isolate around 2,000 fungal strains in axenic culture and establish the Instituto de Química Fungal Collection. Yeah. This collection is taxonomically diverse. We have sampled, we have identified at the genus level around 10% of this fungal collection, showing a high diversity in the taxonomy of these microorganisms. So then to further advance with our research, we fermented around 800 of these microorganisms in several media to map the chemical space that is contained in this fungal collection. So we applied liquid chromatography coupled to mass spectrometry and organized the information into molecular networks that allow us to map the diversity of the chemical space contained in this collection. So the next step in our approach is to evaluate the bioactivity of these fungal extracts. To do so, we have implemented several bioassays in our lab, including inhibition of protein tyrosine phosphatase 1b and inhibition of the growth of bacterial strains and biofilm formation also. And using these assays, we have implemented, we have performed more than 4,000 biological evaluations and integrated this information into molecular networks that have the chemical information about these extracts. So taking all this information together, we were able to prioritize some fungi to perform chemical investigation. So based on these results, we selected Thalaromyces amestolchiae and ant-associated fungus for chemical investigation. So this study led to the isolation of several fungal natural products, secondary metabolites, known as dimeric phenylenones. These dimeric phenylenones display a good activity as PTP1B inhibitors. Importantly, in this study, we use a full construction of protein tyrosine phosphatase 1B, as it could provide us information about its allosteric inhibition or allosteric modulation. So with these molecules in hand, then we next evaluated the IC50 values for every single one of those molecules, and interestingly, the activity was closely related to the structure of the molecules we isolated. So then we evaluated how this molecule, the most active molecule that we will call duclauxin from here after, how it behaves as an inhibitor of PTP1B. We apply some kinetic experiments to demonstrate that this molecule behaves as a mixed inhibitor of protein tyrosine phosphatase 1B, as there is changes in velocity and the Michaelis-Menten constant. Also, we tested the affinity of this molecule to towards the protein using quenching fluorescence experiments, and determined that at that moment this was the most stronger, the compound that showed the most strongest affinity through the protein. So, motivated by these results and taking into consideration, and I think many of you will love this, or would love to hear this. This particular fungus, Thalaromyces amestolchiae, is an excellent producer of duclauxin. We reach yields that about a gram of the compound only with 10 grams of substrate. The substrate that we used to ferment this fungus were Cheerios, the cereal that we eat in the morning. So also motivated by the previous results, we started a following up project in which we modified the structure of duclauxin to get better inhibitors of protein tyrosine phosphatase 1B. So the chemical design was guided by molecular docking. Sorry for the pictures. So after we selected the most promising candidates, we established also a route to synthesize them. The goal here was replace this oxygen atom at position 2 by a nitrogen atom, so we can generate a collection or a library of derivatives of this molecule. Once the molecules were synthesized, all of them were characterized using spectroscopic and spectrometric data, such as nuclear magnetic resonance and mass spectrometry. So, following this approach, we were able to synthesize around 34 examples of duclauxin derivatives, and in the process to get sufficient material to perform the chemical modification, we got 6 additional natural products. So our collection in this kind of molecules is about 40 molecules, and we tested all of them as inhibitors of protein tyrosine phosphatase 1B. So, sorry for the slide, but what I wanted to show in this is that during the purification process of the intended molecules, we noticed also in the chromatogram that aside from the expected peak or the expected compound, we were having another component with a batochromic shift in the UV profile, meaning that this molecule was modified and incorporated an extra double bond into the molecule, which likely proceeds through an elimination of this methanol molecule that is here to form this double bond. So importantly, these results, when we evaluated these molecules as inhibitors of PTP1B, the biological results indicated that compounds in which methanol have been eliminated are in fact more potent than the starting material, and the compounds that we expected or we anticipated will have a good activity. So, I don't think my slides will show correctly, so sorry for that. Well, we then focus our efforts on synthesizing compounds in which this methanol molecule had been eliminated, and got around 20 to 25 molecules of this series, and also correlated the activity of these molecules with some molecular descriptors. The descriptor that better correlate with bioactivity was the total volume of the molecule, meaning that largest molecules display better activity. Also, during this structure-activity relationship study, we noticed that para-substituted anilines behave better as inhibitors of PTP than ortho and meta-substituted anilines. So we synthesized these molecules, and through this process we were able to improve the potency of the starting material by 1 order of magnitude, 10-fold. I think was the best inhibitor. So with these results in hand, then we evaluated the toxicity of the molecules, the starting material, and also what we called at that moment the lead compound. So we evaluated this in vivo models. So both compounds didn't show any toxicity after 14 days of evaluation. And the next step was to evaluate the efficacy of these molecules in an in vivo model to see if these compounds really behave as an inhibitors of PTP1B in vivo too. So we applied an insulin tolerant test in which we evaluated the compounds at 3 different doses, and it's clear that the compound by itself doesn't have an effect on reduction of glucose levels. However, when it is administered in combination with insulin, it lowers down the glucose levels in mouse. So, explaining or corroborating that the mechanism of action of this molecule is through the inhibition of protein tyrosine phosphatase 1B. So, a side project that came from this research was this work or this research that we published in Journal of Natural Products, in which we explored the chemistry of this type of molecules, particularly duclauxin, that was the main product synthesized or biosynthesized by this fungus. So we interrogate if the different derivatives that have been reported in the literature were in fact natural products or were artifacts of the isolation process. Through a very simple experiment that consists on absorbing duclauxin onto silica gel for 1 week and let it there for that period of time, and then performing an extraction and then a chromatographic profile of that extract, we were able to identify at least 6 to 7 compounds that had been previously reported in the literature as a natural products. So through a series of experiments, we concluded that in fact those natural products or those previously reported natural products are in fact artifacts of the isolation process, and many of the duclauxin derivatives reported in the literature are in fact these kind of molecules. So to expand a little bit more the chemistry of Thalaromyces amestolkeae, we started a different project in which we added to the growth culture some epigenetic modifiers. Our epigenetic modifier was in this case dimethyl sulfoxido or DMSO. So there is relevant changes in the chemical profile of this fungus after, well, here is duclauxin, okay? There are significant changes in the chemical profile of this fungus growth under a standard condition, which is this line in black, and this is the major product in that chromatographic profile. So when we added to the culture 5% DMSO, the chromatographic profile changed completely. So we performed chemical investigation on this molecule, on this extract, and we were able to isolate 15 different natural products that are not produced under standard conditions, including 2 new chemical entities. So with this information, I would like to. I hope with this talk I show our broad strategy, which includes natural products chemistry, also biological activity, and some other experiments to, one, explore, understand, and expand the chemical diversity of fungal secondary metabolites. And also, I would like to say that from one single strain, we have been able to isolate, characterize, and biologically evaluate about 66 compounds, and this is only the beginning, because the reactivity of this molecule could be further explored to get more analogs. So with that, I would like to say thank you to the institutions that have supported our research, and also to the people who at one point believed on me, such as Dr. Mata, Dr. Nicolas Overlis, Mario Figueroa, Dr. Jorge Peon, who believed in me and gave me a position as an independent researcher at the Institute of Chemistry. And also, I'm deeply thankful to the people who do the work, my students in my research lab, previous generation, the generation that is right now in the lab. And before I finish, I would like to say something special to very outstanding mycologist, dear friend of mine. So happy birthday, Jose.
Ahorita las preguntas, ¿eh?
Is there any question on Zoom? Nope, no questions. Okay, so we have time for a couple of questions here.
Beautiful talk. Very, very nice.
Thank you.
I'm curious, with your semi-synthesis, how did you turn one lactone into an amide without turning the other ones into amides? Was that hard?
No, it was serendipity, actually. Before we established that synthetic strategy, we performed some osmotic approach. In that osmotic approach, we co-culture Thalaromyces amestolchiae with Klebsiella pneumoniae. Klebsiella pneumoniae produces so many proteases that are degraded, and also during this degradation, ammonia is produced. So the fungus take this ammonia and incorporate it into the structure as a defense for the ammonia produced by the bacteria. Well, that is our hypothesis. So taking that lesson from that co-culture, we decided to prove several amines and try to incorporate them into the nucleus of ducloxy. So that was the key point that tell us these modifications could be done.
Very beautiful. Thank you.
Okay, Dr. Mata.
Pues qué bonito, Pepe. Muchas felicidades.
Gracias.
Congratulations. Very nice talk. Pepe, you regarding your DMSO, you know, we tried because you told us, and I think it depends of the fungal species. I don't know what Josefa think about that, but no, in all the species, you can change the profile. So we did, and it was worse.
It is actually dependent of the species. Yes. So we have tried, I will say, this methodology with about 50 fungi, and it has worked in 3 to 4.
Oh.
So it's work that have been iterated to get the fungus that will respond. Okay. Thalaromyces amestolchiae is a beautiful piece of fungi. So
yeah. Okay. Thank you and congratulations.
Thank you.
That's one. So I have a question as well. So thank you for the talk and about the bioprocess. That is like okay, you have like really good yields, but how you really scale up curious, right? Or what is your thinking on really get this compound into the market or?
Nestle won't be very happy. So the next step in our production of duclauxin is try to use liquid media to produce duclauxin. So we are starting with some trials there, but we haven't results yet.
Thank you very much, Pepe. Sí. Ok, la siguiente presentación es de la doctora Berta Josefina Espinosa Gutiérrez, quien, créanlo o no, fue mi maestra hace 20 años, ni se acordaba que había sido mi maestra de parasitología, aquí en la facultad. Bueno, la doctora Espinosa es una destacada investigadora con 41 años de trayectoria en la UNAM. Es investigadora titular del Departamento de Inmunología del Instituto de Investigaciones Biomédicas con nivel 3 del Sistema Nacional de Investigadores. Es bióloga por la Facultad de Ciencias y posteriormente realizó una maestría en Investigación Biomédica Básica en el Instituto de Investigaciones Biomédicas y un doctorado en Ciencias de la Vida por el Instituto Weizmann de Ciencias en Israel. La doctora dirige el Laboratorio de Estudios en Tripanosomiasis y Leishmaniasis, donde sus principales líneas de investigación se centran en la mejora de diagnóstico para enfermedad de Chagas, el estudio de virulencia en Trypanosoma cruzi y el desarrollo de nuevos fármacos para el tratamiento de la enfermedad de Chagas y la leishmaniasis. Ha publicado más de 90 artículos, muchísimos capítulos del libro Memorias y cuenta con mucho más de 30 publicaciones de divulgación científica. Es activa tutora en distintos niveles de licenciatura y posgrado de la UNAM y ha participado en numerosos programas de posgrado, así como es coordinadora del curso de Inmunología Aplicada en la Facultad de Ciencias. Thank you very much, Professor Berta, and welcome. Las pasa ahí si quiere con el pointer. El de hasta arriba, el rojo. Pero creo que no se puede primero.
First of all, I want to thank to the organizer and Mario and Connor for the invitation to be in this symposium. I need to say that we are biologists in my laboratory and specialists. We are parasites, not parasites, parasitologists. Sorry. And I realized now that I was showing the presentation that I have missing the first slide, the one that I was talking about the diseases and the parasites that we are studying. These are neglected tropical disease caused by two parasites, Trypanosoma cruzi and Leishmania, with 20 different species. And these are considered neglected tropical diseases because they are present in tropical areas of the world and affect a very impoverished people, causing in them many problems in health, economical, and social conditions. These two parasites are one single parasite that they have very complicated life cycle, going from the infective state to the non-infective state, living sometime in the gut of the salivary glands of the vectors, and in other time of their life cycle inside the humans or other mammals that they are infected. The problem with these diseases are that they are having some treatment that are not more effective, and they are just using with efficacy in the acute phase for the case of Trypanosoma cruzi, and is less effective in the chronic phase. Also, it does not prevent the hair damage that the parasite causes, and also have side effects like ones that are dermatitis, neuropathy, some other adverse and side effects. For Leishmania species, we also have problems with the treatment that has been developed, because they also have variability effectiveness depending on the species of the parasite that is infecting. Also, the duration of the treatment are very long, and the individuals stop the treatment because it takes 16 days for the Chagas disease treatment and almost a month in the case of leishmaniasis. Then these are the justification to develop new molecules to treat these diseases. I would like to say that after hearing all the presentation of yesterday and today, I feel a lucky person, because we are working in this field just a few years ago, about 6-7 years ago, and we have already a synthetic molecule that is very promising, because we are working with it in the mice model in the preclinical studies. But in the natural field, we are just having few molecules to study, and I will show you now only 2 examples of the experiment that we are doing with the material that we are getting from our collaborator from the Chemical Institute and other institute in different parts of the Mexican Republic. I'm sorry. The first example of the work that we are doing with a natural product is our collaboration with the group of Dr. Reyes Chilpa from the Chemical Institute, and working also with my student Carla Rodriguez, that were working with this molecule that they were purifying from this tree, Calophyllum brasiliensis, that is found in the humid tropical rainforest from Mexico to Brazil, and they obtained this material from the leaf of this tree. At the beginning, they found that it was a mixture of 3 different compounds, like the one that is showing here, and we call it this ABA, and also we have ABB and another compound that is named ABD. This mixture was obtained by the classical and traditional methods. Sorry. The one that the Dr. Chilpa was doing in his lab, and is, as I said, a traditional, sorry, very sorry. It's a traditional extraction, alcoholic extraction, and then chromatographic test to get a crystal amount of the material that you can see here in the thin layer chromatography, that they got almost a pure material after these procedures. And when they went to do the NRM studies and the HPLC assay, they showed that the material contained a big peak of material, but it was also present to other peaks of this other type of coumarins. But we decided to work with this material because 93% of the material was this ABA coumarin that was almost pure at this stage of the purification process. I will show you just an example of the kind of work that we are doing in the lab. And one of the first characterization that we made was to see if the movement of the parasite is affected by this material and also the growth. These parasites that are very small, but they have a flagell that produce very active movement, and you can see this under the microscope. So we went to see if this coumarin has the effects of on the mobility of the parasite, and as you can see here, it was an affection depending on the concentration. We also went to see if we can grow the parasite in culture after the incubation with this coumarin, and as you can see, we also observed an effect on the growth of this parasite in vitro cultures. One of the characteristics that we are looking for is also the viability of the parasite, and we do this with the MTT assay. And as I showed you here, we have working with the epimastigot, that is the phase, not infective phase of the parasite, and also with the tripomastigotes, that are the infective phase of the parasite. And as you can see here, we got different effect on the viability depending on the stage of the parasite. Here I show you the epimastigot parasite, that is, as I said, with elongate shape and have a flagell, and also and we found that the LC50 was around 30 micrograms per ml, and also when we went to see with the tripomastigotes, they were affected by a LC of 13 micrograms by ml. It's important to check in parallel the effect of these coumarin on the cytotoxicity of cells, epithelial cells, and I show you here our experiment with these cells, and we can observe that these the lower concentration did not affect importantly to the cytotoxicity in this cell, and we also observe that with the higher concentration we start observing some cytotoxic effect on the cell. The CC50 was found in 55 micrograms by ml. That is different and gives us a window to work with these compounds that is affected by the coumarin, but in higher concentration. Sorry. The other important changes that we observed using these coumarins were morphological changes and damage to the parasite in her shape and in the membrane. We'll see this better in the electron microscopy results, but here you can see the control with epimastigotes and tripomastigotes, and after 4 hours in incubation we start looking at the rounded parasite and the loss of flagell. The tripomastigotes were also affected, and here most of the parasites were without the typical shape, and the membrane was also affected, as we'll see. We'll see here in the electron microscopic studies. The parasite has elongate shape and have this special organelle that is called synetoplast, that is very condensate DNA material, extranuclear. But after the incubation, the effect of the coumarin was evident the shape of the parasite was different. We saw a very thin membrane, cellular membrane, and almost all the material of the cytoplasma was outside the parasite, due probably to the excretion of these vacuoles. And at the end, which we can see here also very thin membrane in the cellular parasite, and it was evident that the damage was important for this parasite. We also went to see if the very important, if the ability of the parasite to infect this epithelial cell was changed or affected by the coumarin, and I hope you can see here with the black arrows that are signaling the parasite inside the cytoplasm of these cells. And you can see here the control, and then observe the decrease of the intracellular parasite depending on the concentration that we were using. It is more clear here in the graphic. And we also observed that the effect of the percentage of infection was reduced after few hours of incubation with this coumarin. When we saw that this coumarin can affect the shape, the growth, and the infection process, we wanted to see how was the mechanism action of this coumarin, and we went to do these flow cytometry studies to ask if this process can be a kind of apoptosis in the parasite, and I showed you here some of the results, and in the graphic you can see that the process of destruction of the parasite is a apoptotic phenomena that is observed here, and that is produced by different concentration of the coumarin. We currently we are doing more experiment to see if there is a oxidative stress in the parasite, looking for oxygen-reactive species in the parasite. I will not show you this experiment. We are doing there with this methodology, the flow cytometry, but because of the time, I want to move to the other natural product that we are studying with, and it is antimicrobial peptides that are present in the vector that transmit these diseases. In specific, we are interested in alpha-D defensins. These molecules are part of the immune system. They have broad spectrum of action and can be found in the salivary glands, on the fat body, and also in the hemocyte. They can control the microbiota of the gut of this insect, can help to heal wounds, and also can neutralize toxins. When we were working with the fat body, sorry, I just want to go backward. My student, Paulina Diaz, and the PhD student, Fernanda Arce, were working with these molecules, and they went to ask if they can found this in the insect vector that I am showing here. This vector that I'm showing here is a Mexican vector that we can find in the central part of the country. It's called Triatoma pallidipenis and is a big insect. It can be 2 centimeters long, and it's a very good vector to transmit Trypanosoma cruzi. Paulina was looking at the sequence of these defensins to try to see if they are present in the vector, and we found that there were different types of defensins, defensin 1.1, 1.2, and 4. And then we decided to try to have this defensin in a recombinant product, and she started doing these recombination studies. He got a full sequence of defensin that result to be 1.3, different to the other that we have found before. We used this expression plasmid to produce the recombinant defensin and label it with the 6 histidines. The model of the defensin is here. You can see here the alpha helix, the beta plates, and also in yellow the cysteine that are very important. There are 6 cysteine to form the disulfide bonds that are important for the structure of this molecule. Here I showed you the process of purification, just to show you that we can get very pure material, only one band of about 5.7 kilodaltons, that was also identified by the antibodies against the histidines. We were to see if this defensin have some effect on the morphological changes of the parasite, and as you see here, we can see with the Trypanosoma cruzi some effect in the changes on the shape of the parasite. The parasite is rounded and it's also losing his flagell, and it was determined that it was with an AC50 of 35 micromolar, but we also were interested to see if this defensin can also affect other type of trypanosomatids, like Trypanosoma rangeli, that is not a human parasite but can infect cows and horses. And for our surprise, we also found that this defensin also affect Leishmania major and Leishmania mexicana, with 60% of the cells with a changed shape and 64% for Leishmania major. We also observed at this point that the membrane of the parasite was affected, but we wanted to see better this effect with the electron microscopy, and you can see here the parasite, the normal shape of the parasite, but after incubation with this defensin, the parasite get a lot of vacuoles in the cytoplasm. The nucleus was, the structure of the nucleus was lost, and almost all the material in the cytoplasm was out, and also we observed some of the laches parasite. So the effect of this defensin is very evident at the microscopy studies. We also are working currently, it's very important also to test the cytotoxic activity on mammalian cells, and you can see here that with the MTT assay, no cytotoxic activity was found, and also because it's reported that this defensin can cause hemolysis in the cell, we did the experiments and we found no hemolysis on the mammalian cells. So in this point, we think we have a defensin that have specific activity against this parasite Trypanosoma crucis and Leishmania, and has not effect in the mammalian cells. We also study other functions of this deficit and observe that they affect the growing of Leishmania major and Leishmania mexican, but have few effect on Trypanosoma cruzi. Also, the metabolic activity of the parasite were affected, mainly also to Leishmaniasis, Leishmania major and Leishmania mexicana, and no effect on Trypanosoma cruzi. What kind of cell death we are observing in this parasite? We did also flow cytometry studies, and we can see that the main death is produced by apoptosis in the parasite, and it's also very similar to the other molecule that we were studying. It was observed that the phosphatidylserine externalization was present in this parasite after incubation with the deficit. Well, we have these two molecular natural products that we think can be a promising molecule to do in vivo experiment with the mice model that we have in the laboratory. We have the virulent strain that causes an acute infection in mice, and also we have a chronic infection model for study these products, synthetic and natural. But what is the problem that we have? We have not enough amount of products to test in mice, because we need to do a dozen of infection to prove the effect in vivo, and it takes a lot of time and need more production of compounds, and also we need funding to do this in vivo experiment. It's the same case for the coumarin and also for the defensive. And here is Ignacio Martinez that is helping in all these studies. And in the last slides, I want to thank to all my collaboration, the small group that is working currently at my laboratory, the postdoc and PhD student, and also the funding from PAPIT in UNAM and from CECITI, from the Mexican government. And to you for your patience and attention.
Thank you very much.
Questions?
I learned a lot from your talk, so thank you. I'm curious, with the mouse models of the infections, how long does it take to do those models? Like, how long does it take for the mice to be infected, and then how long do you have to treat them?
With the Chagas disease model, we have this mice model that is infected, and it takes about 2 or 3 weeks to get the acute infection. And then with the not virulent strain, we can go to 100 days after infection, and it is recognized as a chronic model of Chagas disease.
So it's expensive. 100 days of mice is very expensive. Yeah, I understand. Thank you.
I have one question. So with the collaboration of Professor Chilpa, you were able to get a few grams of compounds, or hundreds of milligrams maybe. And if we want to test like fungal metabolites in vitro against Chagas and trypanosomiasis, how much compound we need? Like the minimum.
We were made calculation from the coumarin molecule, and at least to start doing in the mice model, we were asking for 1 to 2 grams of this molecule.
Okay, but in vitro?
In vitro, no. In vitro, we can work with the micrograms. It is not problem.
Okay.
The problem is... sorry. No.
It's okay. So we are running out of time. I think we should jump to the next presentation. Gracias, doctora. I'll do it, sorry. ¿Nos escuchas, Edgardo?
Sí.
Cedric. Ok, bueno, después de este baile extraño, tenemos con mucho gusto vía remota al doctor Edgardo Sepúlveda, quien lo voy a presentar. Él nos acompaña desde el CICESE en Ensenada, y brevemente el doctor realizó estudios de licenciatura en la Facultad de Ciencias y un doctorado en Centro de Ciencias Genómicas, ambos de la UNAM, bajo la supervisión del profesor David Romero Camarena, en temas de recombinación homóloga y reconjugación de la conjugación bacteriana en rizobacea. Tras su doctorado fue a Alemania como becario de la DAD, en una estancia postdoctoral en la Universidad de Tubinga, con los doctores Wolfgang Wollenben y Gunther Muth, enfocada a la regulación en la transcripción de la conjugación de actinobacterias. Posteriormente trabajó 2 años como científico en la Universidad de Illinois en Urbana-Champaign con el doctor Stephen Farran, investigando la regulación de la infección por la conjugación en Agrobacterium y el uso de monóxido de carbono en rizobacea. Debido a un ambiente político y social en Estados Unidos, viajó a Alemania y se incorporó al Departamento de Evolución de Proteínas liderado por el doctor Andrei Lupas, en donde se dedicó a la caracterización de mecanismos de traducción de señales y regulación transcripcional en sistemas de 2 componentes. Con respaldo de la sociedad Max Planck, finalmente regresó a México a establecer su grupo en el Departamento de Biología de CICESE, en donde sus líneas de investigación se centran en la caracterización de una familia novedosa de sistemas de traducción de señales de 2 componentes diversos. Además, mantiene colaboraciones constantes con los productores agrícolas del Estado a través de proyectos de bioprospección bacteriana destinados a fortalecer la industria regional. Muchas gracias, doctor, y lo dejamos con su presentación.
Thank you very much. It's a pleasure to be speaking to you. So to begin, I will start to talk a little bit about CICESE. CICESE is the biggest public research center in Mexico. It was founded more than 50 years ago, and all the researchers that work there, we work in basic and applied research. CICESE is divided in 4 divisions that overall has 12 departments. You can see in the image we range in several disciplines, and I belong to the division of experimental and applied biology. which we have the departments of conservation biology, marine biotechnology, biomedical innovation, and microbiology. In the picture you can see the campus of CICESE, but what you cannot see is that also there's a campus of UNAM and a campus of the Autonomous University of Baja California, which are together making this academic and scientific cluster that fosters collaboration. The advantages of this arrangement, and specifically of our center, is even though it's the biggest public research center, we are a small community to make enough rapid decisions and execute these decisions. We have these active partnerships not only with the ABC and the UNAM, but also with the University of California system, with San Diego State University, and with Scripps. We have several PhD and master programs, specifically the one I coordinate, which is the graduate program of life sciences, where we have 120 students that conduct transdisciplinary research, often focused on natural products. And of course, the peninsula is almost 1.5 times longer than Germany. So it offers a vast territory of diverse ecosystems in which we can work. So this is a picture of what surrounds Ensenada. Ensenada is surrounded of agricultural regions. We have to the north the famous Guadalupe Valley, but we also have more to the California, more to the center. We have Ojos Negros, and to the south we have Mañadero, and even more to the south we have San Quintin, where also a lot of agricultural products are being grown. The main market for these agricultural zones is California, and particularly the organic market of California, which is very, very big. So our producers have a big motivation to produce organically, and this means that they have to adapt the agricultural strategies to reduce the use of agrochemicals. So overall, Baja California has a surface of more than 70,000 kilometers. We have temperate and arid and very arid weather, and 78 different crops are grown in the state. But the main produce is tomato, organic tomatoes, very nice tomatoes that you will never see in stores in Mexico. Tomato cultivation is specifically in California. In Baja California, it's really complicated. It's very susceptible to pests and diseases, and it also has high nutritional requirements. For example, per production cycle, approximately 35 agrochemicals have to be applied to have good yields. Among them, fungicides, insecticides, and foliar fertilizers. It also is prone to suffer for several diseases, like the ones I am listing here. And the one I'm going to talk you about a little bit is vascular wilt, because it was the one that I was contacted by some producers that were having problems with vascular wilt in tomato, in San Quintin, and in Mañadero Valley. Vascular wilt is also in form, especially Lycopersicum, is 1, 2, or 3. This fungi invades the roots of the plant. It colonizes the root, and it causes this necrosis that you can see in the neck of the root, where the place where the plant fuses with the roots, producing this necrosis that not will only impede the movement of water from the roots to the body, but also the movement of nutrients from the upper part to the roots, but will also affect the stability of the plants. So when plants have this disease, they will grow apparently very, very, very good. But then when you have any environmental stress like water scarcity, high temperatures, the plants will start to marchitate. They will start losing the color. Even if they are big enough to start producing, since the structure of the plant is compromised by the necrosis, the plant will just die, and this will affect yield really strong. So in Baja California, the way to combat this kind of diseases is using plant growth promoting bacteria, microorganisms, which are beneficial microorganisms that interact with plant roots, and they promote plant growth, but they also promote resistance to phytopathogens. And there are a lot of efforts and a lot of to develop this kind of biological agents for the agriculture in Baja California. The problem that we have seen is that often the commercial application of PGPRs fails because they just bring strains that were isolated, for example, in Israel, in Dutch, in the Netherlands, and they are not able to establish successfully. Therefore, they are not able to promote the growth of the plants or to control the plant diseases. Why this is happening? Well, this happens because colonies, to work, these bacteria have to be able to colonize and to stay in the plant. And this is affected by several factors, including weather, critical soil properties, the existing microbiome that is very resilient and does not accept newcomers, and of course plant bacteria communication. So often people bring here organisms that were isolated from onions and then try to apply it to Solanaceae. So the philosophy of our group is to use related plants to the crops that we want to treat, and also plants that are native to Baja California. Therefore, we are trying to isolate bacteria that are already adapted to the environmental conditions where the crops are being grown, but are also able to interact with these crops because they are related to the plant that they were originally isolated. For example, for this problem in tomato, we screen bacteria from Solanum hinsianum, which is a Solanaceae that is endemic to Baja California and the Sonoran Desert. It loves to grow in arid conditions. It's resistant to the lack of water, and of course, it's a Solanaceae. So we were expecting this bacteria to be able to chemically communicate with the plants, with tomato also, because they are somehow related to the crop. So to the sampling, we did a very nice travel in the peninsula. We did enjoy, and we do enjoy our trips to the peninsula. We don't have alligators in Baja California. Yeah, nothing. Maybe some coyotes, and that's all. No, not a big problems. And the places are beautiful. The desert is amazing, and well, almost all the travels were done by students. I was only able to do travel often. No? So from the bacteria that we isolated from the soil and the roots associated to this plant, we isolated 343 bacteria isolates, which we screen in microplate assays for diverse characteristics associated with plant growth promotion. And we selected a group of the bacteria that showed the biggest amount, or the more abundant, who seem to have the best profile in growth promotion characteristics, and we tested them in the greenhouse in a very big experiment in tomato. And we were able to identify several bacteria, almost all of them belonging to the genera Pseudomonas, that were able to improve plant height and carbon incorporation into the plant, and also tomato yield overall at the end. No? We have also, in other experiments that we will not show, we have demonstrated that these bacteria are able to colonize and stay in the plants for a long time during the whole agricultural cycle. But of course, we were also interested in the bacteria that were able to combat this Fusarium oxysporum. So we made a specific screening with several bacteria of the genera Bacillus and Actinobacteria against Fusarium oxysporum race 1 and race 2, which were two isolates that we obtained from the crops. These were the real fungi that were causing problems in Baja California. So we made this big screening to try to identify bacteria that will be able to inhibit them. And we selected 8 isolates, among a lot of which we were able to find that were able to inhibit the fungi. But particularly, we selected these ones because they were the one with a higher antifungal activity. 2 of them belong to the Streptomyces genera, and all the other were bacills. We made the experiments to try to see if these plants were going to be able to control the disease. And we were able to find that at least this isolate, BES262, when applied to the plants together with the pathogenic fungi, was able to diminish and to disappear the symptoms caused by the disease, showing that they were being able to control it. Nonetheless, the fungi was still there. The fungi was not being exterminated by the bacteria, but it was controlled to the point that it was able not to continue the production and not to damage the plants. These bacteria are already transferred to the producers, and it's being this technology is already in use by a lot of producers of tomato in Baja California. But during the course of these experiments, we found a very interesting result, and it was with the actinobacteria isolate RBES 331. When we applied it alone in the plant, we saw that only 66% of the plants that we apply the bacteria to the plant survived. 33% of the plants died. But it was even more interesting when we applied the bacteria together with the fungi. In this case, only 33% of the plants that had this treatment survived, which showed that something was happening in the interaction between the fungi and the bacteria that was causing the death of the plants. And you can see in the image that even though apparently these are only one of the surviving plants, one of the 3 surviving plants, you can see that even though the aerial part doesn't look that bad, the root looks really damaged, and it's not normal compared to the other plants with the other treatments. So this brought back my attention to the actinobacteria, which were, I used to work with them for 2 years, interviewing with Dr. Volleven and Dr. Muth. And I decided to start side research with the actinobacteria. I decided to test these 4 bacteria, RBES 331, that is the bacteria that kill the plants, and other 3 bacteria that were shown to have a good antifungal activity. And we tested them against Fusarium oxysporum. I must apologize, the names are mixed. The one in the left is Fusarium oxysporum. The one in the right is Lacediplodia brasilensis. Fusarium oxysporum, as I told you, is a causal agent of tomato root rot, and Lacediplodia brasilensis is the causal agent of grapevine dieback in the Guadalupe Valley. Again, these 2 isolates that we are working with are real isolates that we obtain from diseased plants in the peninsula. So what we did first was to test the supernitants of these actinobacteria. We grow them in different temperatures to try to see which temperature will produce better antifungal activity. We tested 3 different temperatures, 2 different days of induction of growth, and these 4 different strains. We found very good results with isolate 331. At 37 and 40 degrees, it was the one that had the strongest antifungal effect against both fungi. And well, you remember this is the bacteria that also seems to be killing the plants when applied together with the fungi. Then this was a work with a student. We also did we followed the strategy that a lot of you have already found, that is to use inducers to try to change or improve the production of the active metabolites in the bacteria. So what we decided to do was to use fungal supernatants as inductions. So we will grow our fungi for 3 days, and then we will obtain the supernatant after filtration. And then we will add 2.5 milliliters to 5%. We will add the supernatant in a 5% concentration to our actinobacteria cultures, and then we will obtain the induced bacterial supernatants, and then we will repeat our experiments. And here is where we found something really interesting. In the case of strain again 331. We found that when we applied when we applied the supernatant of Lacediplodia brasilensis to strain 334, we will find a decrease in the activity. So compared with the non-induced strain, the supernatant. The supernatant of the strain induced with the Lacilopleura brasiliensis inducer will show a decrease in the production of inhibition in its inhibitory characteristics. And it will happen the same with Fusarium oxysporum. If we will add the culture with the Lacilopleura supernatant, the resulting bacterial supernatant will decrease its antifungal activity, which will not happen when the strains were induced with Fusarium oxysporum supernatant. So we have now this ambivalent situation in which one fungi will induce or improve the antifungal activity of the actinobacteria, while the other fungi will reduce the antifungal activity of the actinobacteria. And of course, for someone that is interested in natural products, probably they will just follow the route of the treatment that will improve the antifungal activity. But since I am interested more in transcriptional regulation and ecological interactions in soil, and we are now following both directions. We are interested in understanding which are the molecules that are causing in each fungi this different response in the actinobacteria, and we will also be looking to understand which are the transcriptional changes that happen in the bacteria when it's subjected to one or other supernatant. For this, we will try to integrate our experimental data in the interactions with fungi and bacteria with RNA expression data, and we will try to identify and characterize the regulators that are involved and the signaling molecules. And of course, this is only an example of what we are doing in the applied research. We have a big collection of plant-associated bacteria, and from these bacteria, we also are building a collection of plant-associated actinobacteria. I already told you the story of Mariola, but we are also working with bacteria associated to desert grape, Vitis giardiana, to get bacteria to work with for the wine industry in Baja California. And we are also working now in a multi-institute effort characterizing and isolating bacteria from Dahlia, which is a Fabaceae, to try to find bacteria for the production of not beans. We do not grow beans in Baja California. We do grow other Fabaceae that could be benefit by the use and isolation of this bacteria. Finally, I still have 1 minute. I would like to thank the persons that did basically all this work and did have the opportunity to travel and do the sampling. Carmen, Anna, Jenny, and Jessica. And with those, this is all what I have to share with you today.
Thank you, Edgardo. Question? One question.
Oh, can you hear me okay, Edgardo?
Yes.
Okay. Thanks so much for giving the talk. Really appreciate you being here. I have a quick question. Have you ever observed that a bacteria or fungus that has a positive effect in the vegetative phase of the plant's growth then has a negative effect in the fruiting phase? Because as I understand it, has different demands on nutrients that it wants to uptake, right?
Yes, we just finished. I have two students in collaboration with Dr. Rafa Bello on Autonomous University of Baja California, and we tested some of our isolates against diverse varieties of chili peppers, isolated from different regions of Mexico. And we have shown, or we have found, not shown yet, that some bacteria are very good to improve the growth of the plants, but this will have a very big impact on yield later. And the thing is that sometimes we humans think that bigger is better, but at the end, what the producers, the farmers want is an increase in yield. They don't care the plants are really big and really leafy. They want this carbon and all this energy to go to the tomato. So yes, there are some isolates that improve so much the growth of plants, that the yield will be reduced. So it's important always to test your isolates. And it also depends on the moment you apply them and how much you apply them. It's very, very diverse.
Interesting. Thank you.
Over the time, so thank you, Dr. Edgardo, for your wonderful talk.
Thank you.
So I'm asking everybody to be patient. Another 30 minutes, 35 minutes, please, and then we will be done soon. ¿Nos escuchas, Lorena?
Sí, ¿se ve? ¿Me escuchan?
Sí, se ve bien. Ok, so nuestro siguiente ponente es la doctora Durán Riverol. Ella es investigadora también adscrita al CICESE en Ensenada y ella es ingeniera bioquímica, seguido de una licenciatura en literatura y una maestría y doctorado en ciencias marinas en el CICIMAR del Instituto Politécnico Nacional. En el 2004 su trabajo fue reconocido por el Premio Lázaro Cárdenas a la mejor tesis doctoral en el área de ciencias médico-biológicas del Instituto Politécnico Nacional y actualmente dirige un equipo de investigación enfocado a la ecología química de microalgas y está a cargo de ceparios de cultivos de dinoflagelados del Departamento de Biotecnología Marina del CICESE. Muchas gracias, doctora Lorena, and you can start your presentation.
Thank you. So please feel free if you don't hear well. I'm going to talk about the marine dinoflagellates as amazing molecule producing machines. And first of all, I want to talk about our collection. We have in this collection mainly dinoflagellates, but not only. And this collection comes from samplings in Ensenada, San Quintín, La Paz, Acapulco, Huatulco, Puerto Ángel, Veracruz, Campeche, Puerto Morelos, and Cuba. So all these strains come from everywhere in the country and also other country, and it's a registered collection. We have some of them who are planktonic. These are the ones that are floating in the surface of the water, and some are benthic. And these are the ones that are attached to other surfaces. And this is the collection of the benthic ones. But what are dinoflagellates? Dinoflagellates are microorganisms, mostly marine, but there are some freshwater too. More than 2,000 species now, and this number continues growing because we found more and more species, and more than 100 of them are very infamous because they are toxin producers. The thing is that we have put our attention in the toxins because toxins kill marine animals and also people, but they are amazing metabolic machinery, and they not only produce toxins, they produce other metabolites that are of interest. These organisms can be photoautotrophic. I mean, they can use only the sun and nutrients for photosynthesis. They can be heterotrophic. They can eat other organisms, but most of them are mixotrophic, and this means that depending on the conditions, they can eat someone else or they can photosynthesize, and they can even eat another organism, steal their plastids, and photosynthesize with them. They can be parasites, and they also can be symbionts, and mostly corals and ctenophores and other organisms. These microorganisms are mostly responsible for these red tides. Harmful algal blooms, and they are not the only ones causing them, but most of the times they are the ones. And you can see red tides like this, but they can also be yellow, they can also be blue or greenish, depending on many factors. They can also be invisible because you can't say where something like this is happening when it's like blue. And they are also responsible for the marine bioluminescence, which is a very nice spectacle if you have the luck to live or to be around a beach when this algal blooms happens. but also they can cause death. And not only fish and snails and mollusks and other organisms, but also birds and also humans too, and marine mammals such as lobos marinos, sea lions, and whales and other organisms. So some dinoflagellates are very well known for their toxins because some of these toxins can cause death at doses of micrograms per kilogram. Saxitoxins, for instance, can kill one 70-kilo person with 1 milligram. And the question we have all the time is, why do they produce these toxins? What for? And we are certain that they don't hate humanity, they don't want to kill us, and these toxins must have some motive. So we don't have clear, and of course every toxin has its history. They could be allelochemicals, they help to fight other algae. They can be defense from predators, they can be enzymatic regulators, and they can also be signaling molecules. Because dinoflagellates, as all the organisms on Earth, they also fight against competitors. And they defend themselves against being eaten or being infected. So they produce cytotoxic and antibiotic compounds, among many others, like antioxidants. And this means that they have great pharmacological potential. But what about their genome? One thing that is amazing about dinoflagellates is that they have an extremely large genome. Not all, but most of them. Up to 185 gigabase in Lingulaulex polyedra, the one that produces bioluminescence. And just as an example, humans, we have 3 gigabases. So this is 60 times bigger. And they can have up to 220 chromosomes. And these chromosomes are permanently condensed. They have lots of unusual bases, and they don't have histones. They have other kinds of proteins. So the metabolite producing, including toxins, this capacity is a result of multiple independent evolutionary events. So they are not linked to 1 or 2 genes, and it's really complicated to identify which genes are involved in this production. So it's not that easy, like, okay, I take that gene and I will put it in a bacteria and then I will produce. No, it's not that simple. So I'm going to talk about some projects we have about the search and identification of cytotoxic bioactivity, some new molecules, some new records, and some new species. Now I'm talking about Culia malayensis, which is dinoflagellate, benthic dinoflagellate, and Amphidinium eliatiense, which is also mostly benthic. So Culia malayensis, this is micrography, a micrograph. You can see it's like a balloon. It's a very, very beautiful organism. But this is how Culia malayensis culture looks like. And you can see this mucus. And if you take a sample and put it in the microscope, you can see how these microorganisms live attached to this mucus they produce. And this helps them to be attached to macroalgae or seagrass or things like that. So, okay, this, when I say we, that's not true. This is one of my students and a whole team. But she was doing this. She used different solvents to get different extracts, methanolic, hydroalcoholic, dichloromethane, and aqueous extracts. And she tested them against some cell lines, cancer cell lines. This is prostate, and this is cervical cancer. And we found very interesting activities on the methanolic, hydroalcoholic, and dichloromethane crude extracts. And the same for cervical cancer with hydroalcoholic and dichloromethane extracts. With this cell line, which is from breast cancer and lung cancer, we also found really interesting activities against them. And of course, as expected, the aqueous extract had no activity at all. But what about the non-cancerous cells? This is fundamental. We want to discover new metabolites that cause no damage or very low damage to healthy non-cancerous cells. And this will be the secondary effects. So we found very interestingly that the methanolic and the hydroalcoholic, yes, they affected normal cells, normal non-cancerous, but not as much as they affected the cancer cells. So she performed lots of chromatography. I'm not going to go into details. Lots of thin layer chromatography too. And then she tested the fractions from this methanolic extract against prostate and lung cancer, which were the ones that responded better. And we found that fraction 4 and fraction 7 of this methanolic crude extract were really potent, really interesting against prostate and lung cancer cells. And all caused cell death by apoptosis. We don't want necrosis. We want to kill them by apoptosis. And they didn't, especially fraction 4, but also fraction 7 mostly didn't damage the normal non-cancerous cells. So these are some of the compounds that she found. I'm not going to go through the whole list, but these are reported anti-cancer, antioxidant, and cytotoxic molecules. And because I know when I talk to natural product chemists, they really love to see some chemical structures. I am showing you the chemical structures we found that this dinoflagellate produce. And they are lovely. They are not too big, and they are kind of complicated, and they have very interesting activity. So if you want more information, I invite you to read her article from Dr. Itzel Morales. And it's for free, and you can also contact me if you want more information. Now I'm not going to talk about Amphidium eliatiense. This is very interesting because we have identified it only in Israel and Mexico. There are only 2 registers of this species in the world. And the Mexican strains have been found to produce several new amphidiol analogs. And what's the amphidinols? Well, I'm going to talk about that in a moment. So this is how the cultures look like. They are very small, and if you take a drop from this and put it in the microscope, you can see something like this. And they are very small. They can be like 5 by 7 micrometers, and it's not easy to culture, but lots of them. And the amphidinol is this structure. It has a lipophilic part and hydrophilic bigger part, and it's very famous and very known from every single point of view. And the Mexican ones, especially Amphidium eliatiensis, they possess the most complex profiles of 8, 9, and 11 different amphidinols, which 3 of them were not reported ever in any other Amphidium strain in the world. And you can find more information in this publication about amphidinols from Mexican strains. So, working with Carmen, I'm sure you know her because I'm sure she is there. She tried this methanolic crude extract of 3 different Amphidium eliatiensis strain against cancer, breast cancer, and she found something really amazing. This MDA-MB231 is a triple negative cancer cell line. This is the most aggressive breast cancer that a woman or a man can have, and they are very difficult to treat, this type of cancers, because they show no response to most of the treatments. And this extract, the crude extract, show a really important biological activity against them. And the lung cancer, we had also a very interesting response, maybe not as interesting with as it was with breast cancer, but what about the normal cells? And this is very important. The normal non-cancerous cells, these fibroblasts, they were not only not damaged by them, but they were growing healthier and more with this methanolic extracts. And remember, this is fundamental because we want metabolites causing low to no damage to healthy non-cancerous cells, but to have a really strong activity towards the cancer of the bad cells. So she went into different kinds of extractions, a non-polar phase, and then lots of chromatography. She's now an expert, a chromatographist. And we chose the 181 because it grew faster, produced biomass, and she fractionated that. And we chose one cell from breast cancer and one from lung cancer for further tests. And she found that the fraction 5 and 6 was very potent against this triple negative breast cancer, and also very potent to this lung cancer cells. And what happened with the normal cells and the non-cancerous, they were almost no damaging or no damaging at all. So she decided, or we decided, to go into a metabolomic approach. So she found the metabolic pathways for chemical compounds, and this is what she found in positive ionization mode. Most of the metabolic pathways corresponded to fatty acids and alkaloids in fraction 5, and in fraction 6, our favorite fraction, because remember, this was very potent against bad cells and not affecting normal healthy cells. And she found that the metabolic pathways corresponded mostly to fatty acids and amino acid and peptides. So this is what we had there. 203 compounds in fraction 5 and 1,400 in fraction 6. So these are very complex fractions to fraction. And in negative ionization mode, we found exactly the same alkaloids and fatty acids and alkaloids and fatty acids in fraction 6, 700 and 1,200 in fraction 6. I'm not going to be very specific about this one, but I wanted to show you some chemical structures. And these are the molecular networks in positive ionization mode. And the molecules, some of the thousands of molecules that were present there that have been reported as molecules with known cytotoxic activities. And in negative ionization mode, we have thousands of molecules, and many of these have been reported with known cytotoxic activities. However, we're talking about thousands of molecules, and most of them have not been yet discovered. And this is very interesting for natural products chemistry, because there are lots of molecules to be found and registered. So for more information, you can talk to Carmen, that she should be there around, and you can access this her paper, Dr. Maria del Carmen Osorio Ramirez. But I want to talk about other projects too, because we like also to play with bacteria and lasers. And this is an AI image, which I think it's a bit, well, it's not exactly what we do. What we do is that we also study the phycosphere. This is the microbiome, the bacteria associated to dinoflagellates, because of course they have a microbiome as all organisms in the planet. So we want to know who are they and what will happen if they are gone or they are changed, like pollution and how this is related to the dinoflagellate metabolism, and how do they change when they are in culture and when they are in nature. But more interestingly, how do metabolites change when the microbiome changes? So let's say we treat with antibiotics the culture, and are they changing the metabolites they are producing? If we put or change different bacteria, how does this will change? And also, we have found that some of these bacteria produce some interesting metabolites, mostly antibacterial metabolites. Also, we have played with lasers, ultra-short pulse lasers, to produce nanoparticles, and in order to improve the photosynthesis and secondary metabolite production through cultures with silicon nanoparticles. And very interesting results. If you want more information, you can contact me. And we are also playing with cryopreservation, because this is not like bacteria that you can just put it in the freezer and let them be there until you need them. We need to make new cultures every month of every of the 300 strains. So we're trying to use some cryoprotectant and nitride, titanium nitride nanoparticles to see if we can freeze them. The freezing process is not the problem, but the thawing process is. So if you want further information, of course, you can contact me too. And I just want to thank all the students, all the team, and thank you. They make me very happy. They are great students and they have great projects, and thank you to all the institutions that are working along with us. Thank you.
Thank you, Lorena. Any questions? Nope.
Well, Lorena, thank you very much for your talk. Again, very interesting to learn about the dinoflagellates, because, yeah, hardly know anything about it compared to fungi and bacteria and other microbes. My question was about the types of molecules that you find in dinoflagellates. When I saw the molecules that you showed, it reminded me a lot of fungal metabolites, a lot of them. Are there any novel scaffolds or dinoflagellate-specific types of new natural products that you would highlight? Or do you think a lot of it comes from the microbiome that they have?
Yeah, no, this is a great question, because it's very difficult to decide which ones are from the microbiome. Sorry?
No, sorry, that was, I don't know what that was. Continue.
Oh, okay. So, but we can have that question when we are checking on fungi metabolites too, because they have bacteria associated. And many people ask this question, and they have treated these organisms with antibiotics, antifungals, and they have found that they are the ones producing them, the dinoflagellates. And they have plenty of unique molecules that are exclusive from dinoflagellates, such as most of the toxins, like, for example, saxitoxin. However, we just were able to put the known molecules that have been found in other organisms, because we had so little biomass to work with that we couldn't make further fractionation until getting pure compounds. And this is the next step.
Yeah, seems like a very difficult organism to work with, but definitely a rich
yes.
source of potential natural products. Thank you very much.
Okay. Muchas gracias, Lorena.
Muchas gracias.
Okay, our very last scientific talk is going to be Professor Cuauhtémoc Lincona. El doctor Lincona hizo una maestría en ciencias bioquímicas en el Instituto de Biotecnología de la UNAM y después hizo un doctorado en la Universidad de Queensland, Australia, con Rob Capon. ¿Rob Capon? Okay. Cerca de donde está el profesor Rob Capon, que algunos conocemos. Y posteriormente realizó una estancia postdoctoral en el Angevio, en el Laboratorio Nacional de Genómica y para la Biodiversidad del CIMBESTAB. Fue profesor del Tecnológico de Monterrey, donde fungió como director del Centro de Biotecnología FEMSA, de Coca, si no me recuerdo, ¿sí? FEMSA, son los dueños de Coca. Y actualmente es investigador en el Departamento de Ingeniería Celular y Biocatálisis del IBT de la UNAM. Lidera un grupo de investigación de innovación en sistemas biológicos, en donde, bueno, buscan instalar aplicaciones de plataformas de biología sintética y sistemas para la ingeniería de microbios. Su labor de difusión se encuentra plasmada en más de 60 artículos internacionales, capítulos de libro, así como una activa participación en la formación de recursos humanos, tanto en la UNAM como en el TEC de Monterrey. Cuauhtémoc, it's all yours.
Okay. So first of all, I have to say that I know that I am in direct conflict with our lunchtime, so I will do this as fast as possible so we can go on it. So yeah, thank you very much for the invitation. First, I would like to show you what one of the projects that we're doing now in my laboratory at IBT in UNAM, and has to do with studying, approaching the discovery of natural products towards the development of a drug. No, hopefully in the next 3 slides, which is a very brief introduction. Yeah, I will convince you that. Well, first of all, here in this slide, natural products have been under development for more than 100 years or a bit more. No. In this timeline at the bottom, I don't know if you can see, we can see that, we can appreciate that during the 60, 70 years ago, there was the golden era, especially of antimicrobials, no, in pharmacy. These metabolites, most of them are inspired on microbial natural products. Or are directly are directly used from microbial sources, right? So most of the structures that were developed, that were introduced into the pharmacy, let's say, were developed long time ago, and at the moment, on the post-genomic area where genomics has been democratized, and as you can see through all these beautiful talks, we can see that. Well, it's now very easy compared to 20 years ago to sequence a genome and to extract information from there. Well, what I wanted to say is that on the post-genomic area, the production or the incorporation of new drugs has been in dramatic decline. There are a few reasons for this. One of them, or a few of them, have to do with the dramatic decrease on investment from pharmaceutical companies or like drug discovery programs, and here in this also slide, sorry, we can see that how genomes have increased the post-genomic area. We know that more than ever we can see that the huge potential that microbes, also plants, both fungi and bacteria, has to give us in terms of new molecules. No, but you can imagine that at this stage there was almost no genome sequences, and a few or a lot of molecules discovered. And at this stage we see that the potential of the biosynthetic in genomes it's huge. No. So, from the perspective of an optimistic microbiologist, like I could consider myself, a drug discovery starts with going to the field. Sorry if the image is not very clear. Going to the field, very happy, bringing samples to the lab, then start isolating, and then from isolation making some cultures in different conditions and under laboratory conditions, of course. and then isolating bacteria and making some bioactivity tests in order to identify if we see that there's a positive or a negative activity, well, it will go through the next stage for compound identification. It's very, it's a bit sad to know that what we are actually, it's the drug discovery pipeline. What I was just talking is just the first 2 to 3 years. And from the time and the amount of money that we need to take one of these nice compounds that we've seen in these beautiful presentations before for myself, we need, well, 10 to 15 years and approximately $1 billion to get one of these compounds to the bedside. For the perspective of an engineer, I am a biotechnology engineer. This is a problem of numbers, and efficiency must be applied. And this is one of the main approaches in my lab. No, techno-economic analyses are very clear, and they say that in order to get one compound to this stage, we need to find in the lab at least 15,000 compounds with positive activity. So I know this is what, at least for me, it's a reality check, a bit sad. But what we're doing in the lab is actually trying to go for, or try to fight for this efficiency problem, no? So this is pretty much what we do in the lab. As you can see, it's like a modified design, build, test, and learn cycle, which is very common for engineer optimization problems. Where we go to the nature. Let's say we bring samples in the lab. We start making some analysis, very basic analysis on genomics, identifying a novelty of compounds. Then we do data integration with other data sets, mostly metabolomics, open profile metabolomics, and from there we start devising a few. I say synthetic biology is not really synthetic biology, but some devices in order to filter all the information that we have built through here, in order to find variable compounds or to pinpoint to where are the actual activities coming from without many efforts. No, this is our sorry. Oops. Yeah. So this is pretty much what we do in the lab. No, we have we would like to go to the field. We like to see genome sequencing, metabolomics, and then getting into mostly Saccharomyces to do some biosensors. All these built around the biosynthetic gene clusters, which are the pathways, as we know, that synthesize the natural products in bacteria and for plants and fungi. So the story what I'm going to talk about now is how we like to sample. Well, Monterrey, as you mentioned, 6 months ago, I was living in the north part of Mexico. So it's very close to the Chihuahuan desert. And there is this a chain of oases that you probably know. It's called Cuatro Ciénegas, that has a very, very specific nutritional characteristics or features in the soil. Mostly, well, there are many, but for the ones that we are interested in as microbiologists, is this disbalance, a very strong disbalance between the nitrogen and the phosphate, the phosphorus source in the soil is completely disbalanced to any other part of the planet. No, so there is a ratio between the nitrogen and phosphate of 157 to 1. While everywhere else in the planet is just 15 to 1. So there's a very, very small amount of phosphate, and this is also, of course, very interesting for microbiologists to go and get samples, and then all the story that I just mentioned a couple of slides ago. We also go to here, I don't know if you can see, but here in the corner of the country, in the rainforest, it's a biosphere reserve that is called Calakmul, which is the biggest deposit of carbon in Mexico, and has also very interesting soil characteristics, humidity, limestone. There's also microenvironmental conditions that are very attractive to go and isolate microbes. So what we do is we go as I mentioned just before, we go, we isolate, we get the genome sequencing, and then a normal prediction of biosynthetic gene clusters, and then crossover with what the metabolites has been they produce under laboratory conditions. So we call this the first step of our approach, this metabologenomics experiments, where we identify and then we start pinpointing candidates, gene clusters or biosynthetic pathways responsible for the metabolites that we see are expressed under a given condition in the laboratory. So the next step, I mean, then it comes the problem, there's a problem with the numbers, because if we have 200 isolates, there will be, if we are a little bit modest, 35 gene clusters per strain. So we would have approximately 7,000 biosynthetic gene clusters or biosynthetic pathways. If we have 1,000 strains, as is in the case in our lab, which is a very small strain collection compared to many other labs in the world, we already have in the lab, or we should have in the lab, around 35,000 biosynthetic pathways. So with all this information, we ask normal questions as an engineer, I would say. What proportion of these biosynthetic gene clusters are truly novel chemistry worth to study? How can we prioritize strains? How, between all the 1,000 strains, which strain would we start to going through experimentation, or start investing money? How can we prioritize strains? It's one of the main questions. And this is, I'm going to show you how we approach this problem. So when you sequence a genome, and then you put your genome sequence in an anti-smash, which is the most common, at least for microbes, way of identifying or screening for natural products, you will get a comparison. You will get a the screening based on cluster families, which I have here definition. Cluster families are defined by protein domains that are common, and then that are common within the whole biosynthetic pathway. With this analysis and with this comparison, and based on something that is called domain sequence similarity, which is based on a Jacquard index, which pretty much what it makes this sequence similarity score, it's accounts. Imagine this family of gene clusters. It accounts how many protein domains have in common in total, and how many protein domains they don't share. So with this matrix, and it's like a distance matrix that it gives you, we devised a very small, a very simple metric that we call it biosynthetic novelty index, BINI, which actually what it makes is you sequence the genome of your bacteria, then you put it in our little pipeline that I'm not gonna go in detail, but based on the distance matrix, the genome size, and the number of gene clusters, we can give you a score of how novel is going to be the repertoire of biosynthetic of BGCs or of gene clusters compared to, of course, a reference. No. In this way, we would have, or we are able to say, okay, we have strain 1, strain 2, only based on genomic information. Which one has the most? Which one, if you are interested in novelty, which one would you start? No, it would give you a score, a quantitative score on the repertoire of biosynthetic gene clusters. So we straight away tested it in our collection. At this stage we had it was 2 years ago, almost 3 years ago. We didn't have a lot of genomes sequence, but we tried with what we have, and we can see that there are a few strains, a few isolates that call our attention. Of course, because of course this has many ways of seeing the problem, right? Because if it has a few clusters, a few pathways that are very well known, and a few others, or a very little clusters or biosynthetic pathways that highly different, well, this would because it's an arithmetic addition at the end of the day, it would have it would compensate, no, the novelty. But it was good enough for us, and we ranked our collection, and we started working on these collections, right? In that paper, in that manuscript, what we were asking questions. I don't know if you can see, but there is a group of bacteria that is called wrongly called rare actinobacteria. It's mostly any bacteria that is not Streptomyces, pretty much. But we went and we collected all the genomes from the most rare actinobacteria. Of course, motivated because we have a few of these in our strain collection. So we and then we ranked what we see here. What you see here is it's the taxonomically classified or taxonomically grouped Actinoechinospora, Lencea, Saccharomyxospora, Saccharotrix, etc., and how diverse was this metric of novelty, no, biosynthetic novelty. And we made a few questions because, of course, we have a group of Streptomyces that comes from Calakmul, a group of Streptomyces that comes only from Cuatro Ciénegas, from this desertic area that I was telling you, a group of Streptomyces that we isolated from plants, and so we also allowed us to ask questions whether the Streptomyces from Cuatro Ciénegas had more potential of encoding novel biosynthesis, novel biosynthetic pathways, compared to, for example, endophytes, or compared to a very well-known or very well-characterized strain of Streptomyces, no? So, so far, this is one of the little stories that I wanted to tell you, using this biosynthetic novelty index. We can rank any strain, and we can pinpoint or prioritize based on the novelty of the encoding biosynthetic pathways. The second story, I am going to hurry a little bit. Yeah, the second story, it's about the development of biosensors, and I like to call it a little bit like non-canonical biosensors, because canonical biosensors, I would like to group them, all the biosensors that are related to growth of, or to kill a pathogen, no? Antifungi or antimicrobial. We are developing and we are working on this type of biosensors, but this type of biosensors, this time, I would like to show you how we prioritize also biosynthetic gene clusters using biosensor. Here is like a little image that we it's under revision in a review that we wrote. So it's pretty much what I showed you before. We have isolates, we test them on different conditions, then we test with a biosensor, and the biosensor will tell us which strain and under which condition we have what biosensor is it's supposed to be revealing, and using genomics and metabologenomics, what I just mentioned, we can identify which of the gene clusters are the most probably responsible for the synthesis of the compound that gets the screening, no, in the biosensor. So the activity that we are screening this time, I'm going to tell you a little background. Aminoglycosides are very well known antibiotics, and these aminoglycosides, of course, they kill bacteria, gram-negative bacteria mostly, aerobic gram-negative bacteria, because anaerobic they are resistant, and in eukaryotes. When you put aminoglycosides in eukaryotes, well, sorry, I forgot to mention that of course, I don't know if you know, but in prokaryotes, in bacteria, aminoglycosides inhibit the protein synthesis. So interacts directly in the ribosome. So in eukaryotes, they also interact with the ribosome, but the activity that they make is, in simple words, if there is a stop codon the interaction of the aminoglycoside with the A site of the 40S subunit of the ribosome will make the ribosome to slip through, to read through the stop codon. That's why it's called codon read-through activity. And this activity is relevant in clinic because there are not a lot, but there are a few monogenetic diseases like Duchenne muscular dystrophy, cystic fibrosis, which are caused by a premature stop codon. In fact, in the 60s, people with Duchenne muscular dystrophy was treated with huge amount of gentamicin, streptomycin, and they were getting some positive results, but they're also getting death, and they're having also horrible side effects. No? So we use this known bioactivity of this type of molecules in eukaryotic ribosomes to develop a reporter system where a dual reporter system where in yeast, in Saccharomyces, which is it has auxotrophy to uracil. So we complemented the auxotrophy of uracil in a plasmid, and we put next to it also a luminescent reporter system. What we did, it was so we inserted stop codons at the beginning of each gene, then and we inserted the canonical stop codon, but also the TAG and the TAA, and then we inserted 2 stop codons at the beginning of the URA3 gene, and then one you can see here what is here, stop codons at the beginning of the luciferase gene. So the rationale is if we have a yeast that is encoding this this plasmid, or one of each of these plasmid, if we put a right amount of an aminoglycoside, it will make it complement the auxotrophy and it will grow on minimal media with the uracil, without uracil, sorry, and same with the luciferase. Well, I don't have time to show you everything, but the stop codon that was the most effective and the configuration was the TGA, the canonical stop codon, and we kept those ones through the experimentation. So here what you see is how the sensor behaves. This is using only the auxotrophic complementation with URA3 gene, and this one is luminescent. So they both work, as you can tell. So if in a culture without aminoglycoside, we used a G418, which is a derivative of gentamicin, as a control. So we tested the sensor without the aminoglycoside and with aminoglycoside, and we can see that after 70, 80 hours, it's growing. It's growing when we have under the presence of the aminoglycoside. In a similar way, in the signal with luciferase, nano-luciferase, we see a different signal when we don't have the aminoglycoside and when we do have it. We kept this one because in this one, after from the second at hour 2 or to hour 6, we have a very nice way of differentiate where we have aminoglycoside and where we don't have aminoglycoside. Once we have this tested, then we tested our sensor with different type of known aminoglycosides, and we saw that well, I don't have the slide here, but structurally this aminoglycoside can have like one aminocyclital ring or a double aminocyclital ring. So structurally, it works only with gentamicin, paramomycin, and of course this G418. No? We tested the range of the concentrations in order to start going into the field and bringing our strains and making our extracts. Well, we had to do like a minimization and to do high throughput on this. I don't have these slides here. But, sorry, so, oops. Yeah, so we went to the lab, and one of each points of here is one of the strains in our collection. Here is the negative control, and here is a positive control of a bacteria, extract of a bacteria that we know produces an aminoglycoside, and the same, that it doesn't produce an aminoglycoside. And we could differentiate, we could prioritize straight away some strains that directly from crude extract give signal into that means there's a presence of a molecule that is similar structurally to an aminoglycoside or an aminoglycoside. Then we made this high throughput, and then we kept after a lot of replicates, because we wanted to validate, especially because none of these green spots here, none of these strains have a canonical biosynthetic gene cluster that would tell us that there is an aminoglycoside in the genome. So we were potentially looking or finding molecules that have pathways that have the same activity, but don't look like a canonical aminoglycoside. Then we did some CRISPR-Cas and inactivated after doing some genomics and pinpointing to a gene cluster that encodes for an aminocyclitol. This is the gene, the aminocyclitol ring. We knock out using CRISPR-Cas this gene, and we see in all our conjugates that we don't have the signal. This is the sensor, the luminescence. For all these mutants, we don't have the signal, which means we knocked out the activity of the cluster that we thought. This is the wild type strain producing aminoglycoside, and this is the positive and the negative control. Anyway, so I think we'll have a minute probably. Well, we are working on this strain, and we are trying to isolate because it's not a canonical compound, and we are chasing this molecule in order to report this. It would be definitely the first natural product that doesn't look like an aminoglycoside that produces a codon read-through activity. As a summary, I'm finishing. Well, I hopefully convince you that biosynthetic novelty index or very simple genomic matrices can be used to identify strains with novel repertoire of biosynthetic gene clusters and compare, and this can be used to compare also diversity across different biomes. The development of this type of biosensors, especially in yeast, using different type of reporter genes with premature termination codons can help us to detect codon read-through activity and diversify the molecules with this type of activity. Well, definitely our working horse is the integration of genomics and metabolomics as a valuable tool for exploring secondary metabolism in bacteria. Sorry. So, well, I would like to thank. I think it's not working. Yeah, the people participating, students that I had before that are all graduated, all the funding agencies, and as you know, I just arrived to UNAM, so I just got accredited as a tutor in all the programs. So I would be very happy if you are interested and you want to talk about projects. I'm definitely recruiting. Thank you very much.
Thank you, Gautama. Questions? One or two?
We're very hungry.
Thank you very much for your presentation. Just a quick question about how artificial intelligence could help in the pathway from the discovery of a molecule and the reaching the treatment. Is there any thought?
Yes, definitely. There is well, the question is if how artificial intelligence has been integrated into the drug discovery, and there are recent tools that incorporate this type of well, methodology, let's say, We are in the lab actually not yet working on this because we are trying to work on novelty, no, like taking the databases and try to look for this the black space, no, that we don't know. So I'm not saying they're not useful. I'm pretty sure there are, but we are like approaching it in a different way. But yeah, there are several tools that incorporate already artificial intelligence into discovery of molecules.
Thank you. Pretty good job.
Thank you, Gautamo. Okay, so this is time for the closing symposium ceremony. Everybody's hungry, I know. So I'm gonna let Dr. Iracema Chirino, which is the director of UNAM Alemania, who is gonna give us a couple of words regarding the program, and she actually helped a lot to make this event happen. And please, Iracema, I don't want to say much about you because you're a very good friend of me, and just I'll let you say some words. Sí, se va a abrir completo. Ahí está.
Thank you very much for the introduction, Mario. First of all, I would like to say that I was here at this place so many times as a student long time ago. So I am very happy to be here again. And I must say that I was here for examinations, for departamentales, if you know what I mean. So I am really happy also to see one of my best teachers, Dr. Rachel. I was a student of Dr. And okay, just it's a very brief presentation about telling the story about this meeting. And this is the title of the presentation because one of the main reasons to be here is fostering the cooperation between Germany and our university. And just to give some details about Germany office, which is located in Berlin. Here you have the map, and in this district, Stieglitz is where we have the office. We have our university has 13 branch offices all over the world, so we are one of the 4 that are in Europe, and this is the International House, is the building in which we are located. Here, this is our office. This is part of the Freie Universität. We are hosted by this wonderful university since 2018. And the mission and vision of our office is to foster mobility, international cooperation, and this information that I'm not going to read, and of course promote these sort of scientific events. And just to give some examples of how do we promote international cooperation, we have been doing a few events in all disciplines, not only in life sciences, but also in the social sciences. So here we have some of the faculties and centers that we are working with in the last year. This event was in the Facultad de Contaduría y Administración. We also are trying to build a cooperation with some agencies and also with the Mexican Embassy in Mexico. This is the building of the embassy in Berlin. And in this year we will have a concert, a piano concert with one of our members of our university. Dr. Monique Rasetti is a full-time professor here in music faculty. So this is also a kind of events that we are trying to foster and to promote. And
And some others, for example, we will have in the embassy, but also in Bielefeld University, short films that were developed by students of the Escuela Nacional de Artes Cinematográficas of our university. So just to give a view of what we are doing. And of course, this meeting, this is just a list about some agreements that we have with several universities in Germany. We are very happy to all of this. And here you see the Gießen University that we are now have our invited speakers. And I'm not going to give some details, but this cooperation with Gießen University is about student exchange, also scholar exchange, promote some different type of research projects. And this year we had 2 students from our university in Gießen. And I would like to just to give some details how we met Connor. So we met Connor last November in the Berlin Science Week in the Mexican Embassy, and this picture was taken before we were talking. So he was one of the winners of a prize, giving a really great presentation of the project that he wanted to develop in Mexico. And after that, we were talking about where could be the place to have this meeting. And as he is from the natural product department at this university, of course, I thought in my colleague Mario Figueroa and all the group that he has been working with, also Doctora Rachel Mata. So I put them in contact, and then we had an online meeting, and then we start to see what can we do together. And so this is the result of that meeting, and I'm very happy that we finally have them. Not only Connor, but also 2 other colleagues, Nicolas Reyes and Alex Skinner, that we are also, they are also here with us. And this is just a summarize of the GEMEX-NAT event that we have. I hope that you all that were participating see your names here, because all together are building this network. So of course this is the topic, natural products, but we are doing international collaboration, and see you here some of the broad topics when we are talking about natural products. I am not an expert, but of course you are, and you see that we were trying to cover all the angles possible to build this network, and I am very happy that we succeed in this meeting. And well, that's it. So thank you very much for your attention.
Well, thank you very much, everybody. So this is the end, but just for this meeting or symposium. So I hope we can get more collaborations. Food now and then collaborations. And I'll see you guys, the grad students that are gonna join the workshop at 3 p.m., 3:30. Maybe we can do it a little bit later. Don't worry about it. And thank you everybody. It's Building H. Remember, it's Building H. It's close to the Mario Molina building. So take your time. We can start a little bit late. Don't worry about it. So see you later, guys.